everybody, good evening and welcome to your very own BYJU'S 9th and 10th grade channel. I'm your teacher Aishwarya, and as a part of Crash Course 2.0, we are going to be doing the entire crash course for the chapter in biology, one of the most important chapters, that is fundamental unit of life. So I hope all of you are excited for today's class, and a very warm good evening to everyone here. Now, I know that a lot of you are very new to our class and some of you are very regular to our class. So for those of you who are new, I'm just going to take 2 minutes of time to quickly introduce myself. I'm Aishwarya, who will be teaching you biology along with Ankita ma'am, and I help you out with SST as well. And in BYJU'S 9th and 10th grade channel, we currently have the Crash Course 2.0 series going on, wherein across subjects, that is your maths, your science, your SST, we are going to be covering all the chapters in this crash course. Now, what can you expect in the crash course? In our crash course today, we are going to be doing all the chapter summary. Yes, we are going to do the chapter summary first. Then a total of 13 important questions are going to be there, out of which 15 questions are going to be subjective, and the 15 later questions are going to be objective, and we are going to be doing it on Menti, right? And all of you I have a checklist with me as well, and as and when we complete, we'll put a tick mark and we will go. Yes, Menti is there, but Menti is the third thing that we will do today. And we will end our session with some tips and tricks that will help you study this chapter well. So everybody, please very quickly before we get started, as you know what we have in store for you today, trust me, this is going to really help you, okay? Especially with your examinations coming up, and you know, right, even 9th grade examinations are super duper important. This forms your basis to learn for 10th standard, 11th, 12th, right? So take your examinations very seriously. Now in class as well, I need your focus to be there, right? So I know that we tend to digress thinking, "Arey ma'am, it's only 9th standard, 10th mein dekh lenge." Don't do that, right? So everybody very quickly hit that like button. We need to get started with 100 likes, right? So quickly everybody, I can see 100 plus live watching is there in my class. Everyone fatafat hit that like button. Those of you who are new, make sure that you subscribe to our channel, because as a part of Crash Course 2.0, we have a proper schedule planned out for all of you, right? So as you all know, today is going to be fundamental unit of life, and that is the 4th session that we are conducting. Tomorrow you have geometry, of course, followed by force of laws and motion on Monday. So as you know, you have the whole planner all the way till Jan 30th, right? As a matter of fact, not just Jan 30th, till February 17th, we have told you what we are going to do, and we will put a tick mark against our session today. So everybody, this is available on our community post as well. So you can go ahead, take a printout, put a sticker in your wall, keep ticking, because with this crash course, right, Crash Course 2.0 is going to be a 3-hour long session, 3 to 4 hours, right? And we give you end-to-end preparation of chapters. And with this, trust me, you will be able to prepare for all of your exams. Hello, good evening, Anshu. Good evening, Alka. I am very good. Have we hit 100 likes? Have we hit that to get started? Shiva, where are we at? I can see Roshandeep, Fatima, Ria, Aditi, Pragati, Pranya. Good evening, Kartike, Niksha. Hello. Yes. Hello, everyone. Good evening. And very quickly, everybody, is my audio, my video, and my screen and what I've been writing on the screen visible to all of you? If it is, give me a quick thumbs up on the chat. Give me a quick thumbs up and we shall get started. Okay, quickly we will get started. Good evening, everyone. Good evening. I'm waiting for just some more time for all of our friends to get started. So I will start very soon. Don't worry. Yes, very good, very good. Shiva, how many likes do we have on the video? 98 likes, 2 more likes, and we're going to get started with 100. And I can see constantly we will increase our like target to 150. And then 200 and so on and so forth, right? So everybody, do not forget to hit that subscribe button. If you are somewhere thinking, ma'am, why should I subscribe? Trust me, 15 minutes into this class, you will then definitely know why it is important to subscribe, right? And lots of love to everybody out there who is sending me, hearts and loves, right? So let's get started, everyone. So here's a quick reminder. What can you expect in our class? So first thing we are going to do is to have our chapter summary. Okay, we're going to quickly recall the important topics from fundamental unit of life. Then we are going to look at 15 questions which are subjective, because your examination is going to be subjective. As much as you love playing in Menti, you should also know how to write answer. Third thing you need to do is to look at MCQs, objective questions, and it will be on Menti. And the fourth one is going to be 5 hacks to study. So as and when we complete this, we are going to put a tick. And everybody, very quickly, Karthik, Tishu, lot of other people who are having parallel conversations in class. See, there are a lot of students here who are learning live to understand the chapter. Kindly do not distract the class, otherwise our moderator will block you. So please stay in class and stay attentive in class. This is all that I ask. Because 2 hours you dedicate with me, trust me, you will be thorough with this chapter. Okay? All right, very good. So all of you have your notebooks and pens. Shall we get started? Notebooks, pens, textbook, everything is there with you. You should have these things, right? Yes, time to turn on focus mode. Okay, we are turning on our focus mode. We are having everything with us and we are going to get started. Yes. If you don't have your textbooks, notebooks, pen, go and get it. Okay, everything ready? Let's start. So we're going to first start out with learning about this particular chapter, cell, right? Now, when you talk about cell, we know that most often than not, we define cell as the basic unit of life, or we say that it is the structural and functional unit of life, right? And why are we calling it as the basic unit of life, or why are we saying it is the structural and functional unit? Because we know that all living organisms that are there, right, all living organisms are made up of cells, right? So cells are like the building blocks. Just like how a house will have a brick, we see that our body has cells, right? And it's also known as the fundamental unit of life, hence the name, right? So now, how did we know that cell was what our building block was? Who discovered cell? Now, this is easily a 1-marker question to 2-marker question. Who discovered cell and who coined the term cell? Now, we know that discovery of cell was made by Robert Hooke, right? Now, can you tell me what did he observe? He observed a certain specimen under the piece of microscope, right? What did he observe? Mrigendra, stay focused in class. I will catch you if you are talking unnecessarily, okay? Exactly, he observed some dead cells. But what did he observe? He observed cork, right? So cork is nothing but the outermost covering of trees, and he observed the cork cells and they were dead cells, right? And it reminded him, it looked something like this, right? And it reminded him of the cells or the rooms in a monastery, which is why he called it, the term comes from the Latin word cella, which means room, right? Hence the term cell. So in this case, we also see that, it reminded him of a honeycomb-like structure. Now, then we have Anton van Leeuwenhoek, right? Now, Anton van Leeuwenhoek, who was there, had a very important role to play in improving the microscope, right? And he was the one who discovered free living cells, yes? So, main point of difference between what Robert Hooke observed versus what Anton van Leeuwenhoek observed was one was dead cells, while Anton van Leeuwenhoek discovered living cells, right? Then, of course, we had contributions by other scientists like Robert Brown and Purkinje, right? So, what did Robert Brown discover? Very quickly in the chat, what did Robert Brown discover? What did he discover in 1831? He discovered a very important thing, a very important part of the nucleus. Exactly. He discovered nucleus, right? And then, of course, we had Purkinje, who termed or who coined the term protoplasm. What is protoplasm? Ma'am, protoplasm, cytoplasm में बहुत confusion होता है. What is protoplasm, cytoplasm? I will explain this in just some time, right? So this is all about cell history. And for those of you who are new to the first time asking me to speak in Hindi, the thing is I am not very fluent in Hindi. My way of communication is more effective in English, right? So the main thing that I'm going to do will be teaching you in English, because at the end of the day, when you are writing your exam, you will not write it in Hindi. You will write it in English, right? Which is why try to stay focused. But if you're not at all understanding, ma'am, कुछ भी समझ में नहीं आ रहा है, I will try my best to explain that part in Hindi. But understand that I will mainly teach in English. Okay? So coming back, alright? So coming back to this particular cell history. The next thing we are going to be discussing is on cell theory. Now, cell theory that was there was proposed by Theodor Schwann, Matthias Kleeden, and later on we had additions by Rudolf Virchow, right? So what was cell theory? Everyone, cell theory they can ask you as well. So cell theory says that all living organisms are made up of cells. So if you have if you have your textbooks, that is there, right? If you have your textbooks with you, just make a mark of it, or if you have your notebooks, you can write it down with me. So all living organisms are made of cells, right? Cells are structural and functional units of the organism, right? And we also know that all cells arise from pre-existing cells, right? And this particular point that all cells arise from pre-existing cells was given to us by Rudolf Virchow. This was a point which was added to the cell theory much later. So this was all about cell theory. Now that we understand, right? So earlier this is how they were learning about cells and they were going about it, and then they realized that cell was the basic unit of life and cell is the structural and functional unit of our life, right? Now, why are we saying that cell is the structural and functional unit, or why are we saying that cell is the basic unit? It is with respect to the functions that the cells carry out, right? Now, cells carry out functions which are essential for the organism only to survive. Because we know there are certain processes that happen within our body that is necessary for survival, right? For example, getting food, producing energy, right? And of course, making sure we're getting rid of waste materials from our body, transport of all of this. Now, we see that all of this is done by cells, right? So, we see that cells have the ability to take up nutrition. Now, what happens is that the nutrients, the food that we eat is broken down and all these nutrients are transported to each and every cell. And this cell that is there will take up these nutrients and they will utilize it. So whether it's in the case of proteins, which is needed for bodybuilding, right? Or whether it's in the case of glucose for producing energy. All of these processes happen within the cell. And whatever waste material is there is quickly given out by the body. So basically, why is cell the basic unit of life? Whatever functions that we carry out is actually carried out by the cell, right? Which is why it is called as the basic unit. So are we clear? Are we all clear? Ma'am, how is a cell formed? See, the first cell was formed by the coagulation. When many of these inorganic components came together, it formed the first cell. But from the first cell, we see that every new cell arises from the pre-existing cell, right? So this was all about cell history and cell function. Take a quick screenshot of this because I'm going to erase some of these pointers. So in case if you want to write anything down, please make a note of it, right? Yes, hello Sukhbir, hello. Take a screenshot, give me a thumbs up, and I'm going to move on to the next part, all right? Ma'am, in human body, every part is formed by cells. Yes. If you think about it, you look at maybe your hands, your legs, your skin, liver, all these parts are made up of cells, right? Very good, okay? Very, very good, everyone. So now let's move on to the next part, okay? Now that we know that these are what are cells, do you think that there is only one type of cells? Do we find only one type of cell in the whole world? Every single living organism is made up of only one type of cell. Yes or no? Yes or no? Very quickly in the chat I need to see. Yes, exactly. No. We see that there are different types of cells, right? So we see that there are different types of cells, yes, which are present. Now, why is it that there are different types of cells? And we see that each of these cells are of different shapes and they are of different sizes. So this is a very common question that they will ask you, especially for maybe assertion reason they'll ask you, or they will ask you as a justify question. Why are there different cells of different shapes and different sizes? What is the reason behind it? For those of you for whom the screen is not clear, go to settings, advanced settings, increase the video resolution. Exactly. Very good. Make a note of this. Different cells have different shapes or sizes so that they can carry out specific functions, right? So when you talk about an RBC, this is discoid, or we say it is biconcave. Now why is it discoid or biconcave? So that at the end of the day, we see that this is able to transport oxygen, right? So that is why it is necessary. Yes. Atoms. Ma'am, atom is small or cell is small? What is atom? You tell me first. Atoms and molecules, you've just done revision. You tell me what is an atom and then you compare it to a cell. You will be able to understand, right? Yes. Screen not clear, bacha. Increase this one. Discoid means disc-like, okay? Ma'am, bone is also made up of cell. Yes, bone is also made up of cell, right? Exactly. So everybody, with this, what do we understand? There are different shapes, right? So we see that different types of cells which come of different shapes and different sizes. Exactly. Atoms are smaller. They're the smallest particle of matter. So technically, atom will be smaller, right? It will be smaller than a cell. Okay? Exactly. It's the smallest unit of an element. Now we know element like nitrogen, you have phosphorus. Now all these things come together and they make up a cell. So atom is smaller, right? Okay. So now with this we have understood this point, and one thing to understand is that size of the organism does not matter. Size of the organism does not correlate with size of the cell. If there is elephant and there is rat, does not mean that elephant has bigger cells, okay, than the rat. Just that elephant has more number of cells. And based on this number of cells, we see that we have types of organisms. So this is based on number of cells. So based on the number, we see that we have unicellular organisms and bicellular, right? So unicellular organisms are those whose full body, okay, their full body is actually just one single cell. Now, can you quickly give me an example of this? Can you quickly give me an example? Yes? Ma'am, what is biconcave? Biconcave means it's a structure like this, right? So you learned about concave, which is bent this way. This is what is biconcave structure or discoid structure. Haploid, diploid, I will tell you when I am teaching you. Centrosome, I will explain when I come to cell organelles. I will tell you about haploid, diploid when I talk about nucleus, okay? Till then, everybody, whatever I am teaching, if you have any doubts, you tell me, okay? Yes, very good. Amoeba is an example. Paramecium is an example. Very good. Yeast is an example. Well done. Now, multicellular means that their full body is made up of many cells put together. And quick examples of this. Quick examples. Yes? Very good. Human being, cat, mouse, anything. Euglena is unicellular, okay? Plants, animals, yes, that forms your multicellular organisms. Now, in multicellular, one thing you need to understand is because there are many cells, we also obtain that, or we also observe division of labor, right? Which means that we have a certain set of cells that carry out a specific function. Not that all cells do all the function, right? So that is what you need to understand. And yes, what do you say? I am also a multicellular organism. Which is the smallest cell? Smallest cell is mycoplasma, right? Smallest cell ka example is mycoplasma. Cell is made up of atoms. See, what is an atom? We know that atom is the smallest unit, right? And in an atom, so if you say oxygen atom, or if you talk about, nitrogen atom, you see that that is one unit that is there. Atoms come together to make up molecules. We have atoms and we have molecules, right? So all these things come together and they make up the cell, right? So that is one way. Which is the first cell? First cell is said to be, something which was formed much earlier. It was a coalesce that was there. Largest cell is ostrich egg. Yes. PPLO is a kind of mycoplasma. All right. PPLO is nothing but a kind of it, of mycoplasma. It is just a type of it. Okay. All right. Yes. Are we clear? Are we clear? Yes. Okay. Longest is neuron in our body, right? It is neuron. All right, everyone. So you can quickly take a screenshot of this. Take a screenshot. Okay, very good. Yes, please side, ma'am. I'm side. Now you can take. Longest cell in the world. Actually, I don't know. But nonetheless, longest cell in our body that is there is nothing but the nerve cell, right? It is nerve cell. Okay. So now let's move on to the next part, all right? Now the next part that we're going to be looking at is the components of cell. So can you all tell me what are the broad components of the cell? Yes? What are the three most important components of the cell? Yes? Quickly everyone. What are the components? Cell membrane, very good. So we have cell membrane. Then what do we have? Cytoplasm. And then of course we have nucleus, right? So cell membrane, cytoplasm and nucleus. These are the three components which are there. Now these components are very, very important as well. Now when we talk about this, right? Especially when we talk about type of nucleus, we also see that we have two other types of cell, which is prokaryotic cell and eukaryotic cell. But at the same time, we see that prokaryotic and eukaryotic, I will cover it better when I talk about nucleus. Now, someone asked me a very interesting doubt, right? Ma'am, isn't DNA the longest cell? DNA is not a cell, right? DNA is an organic molecule, or I would say it is a molecule. It's a, what do you say, component that is there, which is made up of different molecules put together, right? It's a chemical thing if you look at it technically. But that in itself, and that's the most wonderful part about the cell. Cell is made up of many organic and inorganic components, okay? There are a lot of components which come together. Individually, if you take them out, they are all non-living, right? If you take out phosphate, phosphate is a chemical thing, it is non-living. If you take out carbohydrates as it is non-living. You take out proteins, it is non-living. But you come and you put it together when they work, they make it living, right? Yes. Very good. What is DNA, RNA? Everything we will cover in chronology. Ma'am, ovum cell is the largest cell. Ovum is the largest cell in the human body. But altogether, if you look at it, it is ostrich egg. Okay, difference. Ovum is in our body. In the case of ostrich, overall it is ostrich egg. Mycoplasma is a type of bacteria that is there, which is very small. Surbi, Nikhil, badal gaya, abhi class mein focus kar lo. Okay? Yes. Now stay focused. All right. Now everything related to this I am going to come, but first we are going to be talking about cell membrane, right? So cell membrane is also known as plasma membrane, and this right here is a very, very important topic, right? Very important topic. Now everyone, I request you to do one thing. Ask me doubts with respect to what I am teaching now, right? Because I am going to cover all of the topics again and we are going to do practice also. So don't worry about it, okay? So now this is also known as plasma membrane, right? And we see that this particular plasma membrane forms the outermost boundary, right? It acts as a boundary and it encloses all the cellular components, right? So just like how every house has walls, right? What is the job of the wall? It is to make sure that whatever is inside the house is kept there, right? Which is why we also have the cell membrane, which acts as a wall, or it acts as a boundary that encloses everything. Now at the same time, when we talk about the plasma membrane, let's talk about its structure, right? What is the structure of plasma membrane? You all have been telling me. What is the structure of plasma membrane? Yes? Very good, very good. Exactly. It is made up of phospholipid bilayer. So we see that the structure here is when it is made up of a phospholipid bilayer. Okay? So let's understand what is this phospholipid bilayer. Now, this is one single structure. Okay? So what you see right here is a single phospholipid, okay, which means that this part which I've drawn as, you know, a circle, this right here is the phosphate group. All right, so this is phosphate, like chemical nature, phosphate. Now at the bottom, when you see, you have a tail like structure here which is called as the lipid tail, right? So this is the lipid tail that is there. So phosphate head, lipid tail. Are we clear with this so far? Are we clear? Give me a thumbs up in the chat. SER, bacha, I'm going to do. Just stay focused. Fatima, I'm going to answer your question. That's a very good question. I'll answer it. Okay? Yes, exactly. It looks like matchsticks. Exactly. So now what we observe is that there is a bilayer of this. Okay? So now let's visualize this. So we see that there is one of this manner. Okay? And then we see that there is another layer. Okay. Now what you need to focus on is how they are arranged. Alright. So all the phosphate tails are inside, or they're projecting inwards, while this heads are on the outwards. Right. So this whole thing is your plasma membrane. Okay. Now on the other hand, when you see, the reason why they are arranged this way is because the phosphate. So the phosphate head that is there is water loving. Okay? Because we know that most of our body is made up of water, right? So we see that they are made up of, they are water loving. Or someone was asking me, ma'am, what is hydrophilic, right? What is hydrophilic? Hydrophilic means water loving. While on the other hand, we see that on this side, inwards are the lipids, right? Lipids, best example I can tell you is oil. Oil is a kind of lipid, right? Now in this case, lipids are such structures, fatty-like structures. Now these guys hate water, right? So they hate water, which is why this is hydrophobic, okay? So first main component that is there is mainly phosphate bilayer or phospholipid bilayer. But there's not only this. We also see that there is proteins as well, right? Now we see that there are proteins which are present on the outside, some which are found in the middle, some which extend across like this. So someone was asking me, ma'am, extrinsic and intrinsic, right? Extrinsic is which is present on the outside, external, right? That means extrinsic, okay? While which are present inside, right? Or which are there within, which are present on the inside of it, we call it as intrinsic. Yes? Then which are across the layer that is there. Yes, in this case we call it as them as integral proteins or transmembrane proteins, right? So we call them as transmembrane proteins. Yes? So why do they have so many proteins? Because if you see, if you remember later on when you were learning about hormones and everything in your lower classes, right? We know that there is a lot of structures which are there. See, proteins are one thing, but we also have carbohydrates also. There are glycolipids, there are a lot of carbohydrates, lot of components, but we don't need to go too much into the details of it, right? Water loving and water hating, exactly. Water loving means that they will not repel water, right? So we know that there are some which can repel, or they will, you know, not be, what do you say, when you look at water and lipids or oil, you know how they are, they'll not mix properly, right? Which is why we see this way, hydrophobic and hydrophilic. Yes, ma'am, how will it, very good question. Ma'am, if this part of it, right, is made up of water hating part, how is it that water is able to flow through? Yes, how is water able to grow? Because we see that there are certain channels through which water will move, okay? So there are certain channels or certain places through which water will move, which we call it as aquaporins, right? So that is one thing. Yes, this is not there in NCERT, but some of you are asking me questions, which is why I took this part, okay? Understanding for in your exam, what should you know? You should know that structure has phospholipid bilayer and proteins are present, okay? This much is only that you need. Are we clear? Are we all clear with this? Structure, are we clear? Ananya, I am going to get to osmosis, bacha, don't worry. So quickly to tell you, there is a phospholipid bilayer which we observed, which has a phosphate head and a lipid tail. It is arranged like this, right? So this is my phosphate head. The lipid tails are arranged inwards, and along with that we see that there are proteins, right? So there are proteins, and we also see that there are other structures, carbohydrates, which make it, okay? Yes, all right, very good, very good. Okay, yes. So now let's move on to the next one, right? So now we know the structure. The next thing that we need to understand is the function. What is the function of this particular membrane, right? So plasma membrane allows, right? It allows the entry and exit of only some materials, right? So we see that it only allows the entry and exit of some materials, and it denies the entry, right? Which is why we say that it is selectively permeable, right? So it is selectively permeable in nature. Now everybody here, how many of you are confused with ma'am? Selectively permeable, right, versus semi-permeable. How many of you are confused with this? Ma'am, difference between these two. Selectively permeable, semi-permeable. What is the difference? Yes, very good, very good. A lot of you like, ma'am, me, समझ में नहीं आ रहा है. Selectively, semi, both of it is same, right? Exactly. Now, two questions. What is selectively permeable and why should the plasma membrane be semi-permeable, right? Both are not exactly the same, right? Now, selectively permeable means, you look at this word, it allows the entry. It is selecting, right? Okay, think about it. You have an ID card. I'll give you an analogy, okay? You have an ID card which tells you that you are from this particular school and this is your face, right? And because you wear this ID card, you are able to go to school, right? So you are able to enter into school. Now, this is what we call as selectively permeable, allowing only select substances to enter or exit, right? So, plasma membrane here is selectively permeable. But semi-permeable means that maybe due to size of the membrane, right, or especially in the case of osmosis, when we discuss size of molecules, right, and size of membrane matters, which will allow only certain substances to pass through, right? So there it is not allowing. It is because it's like as good as, okay, that is one small door, how many people can fit into it? People who can fit into it will be able to move, but the others may or may not be able to do, right? Which is why based on this we have semi-permeable. Just that plasma membrane is also semi-permeable, okay? That's why normally selectively it will judge, semi-permeable it will go based on size or various other parameter. So that is what we mean by it. And it's just that the plasma membrane can function as semi-permeable also, okay? Very simple and easy. Now somebody was asking me, ma'am, why should selectively permeable membrane, or why should it function as a selectively permeable membrane? Because we are talking about our cell. Now it's not always that good things will enter into the body or into the cell. There could be some harmful things. There could be some unwanted things as well, right? Unwanted things that could enter into the cell, which is why we see that it is selectively permeable. Now moving on, we know that even exit, or we want all the important things to be within the cell, and we want to make sure that only what should leave the cell should go out of the cell, right? Which is why it is selectively permeable. So now are we clear, everyone? Are we clear? Yes? For what is vacuole, what is DNA, I'm going to cover it in the future, Bhavya. Please stay tuned, right? Please stay tuned. So, now Nagda has a very interesting question. Ma'am, how will they know which is necessary? That is why those proteins are there. They will be the ones who will tell, yes, this is needed, this is not needed, various other structures are there. Diffusion osmosis, I am coming to it. Ma'am, can you tell how any disease affects the cell? See, viruses, when they infect, right, they will use the cell mechanism to divide. And that way what we'll do is these viruses will end up destroying the cell. So viral diseases are most often than not is there. Ma'am, plant cell, it is not the outermost boundary. No, it is not the outermost boundary. It is a cell wall, right? Okay, very good. करने से पहले जो आपने लिखा होता है वो साफ कर दिया करो. ठीक है, all right, done junior world. Okay, yes, so now that we have understood the function, right, now let's understand a little more about what are the different ways in which we observe movement. Now we are saying that there is entry and there is exit. So what all things are entering and exiting? Can you tell me in the chat very quickly what is entering and exiting? Quickly in the chat. Yes. Saroj, you can quickly rewind back and see the explanation for this. Okay, you can rewind back and see the explanation. Very good, very good. Yes. Oxygen, okay. Water, yes. What else? Nutrients, very good. Carbon dioxide, okay, wonderful. Right, so these are all the things, right? Now the thing is, for entry and exit to happen, right? For entry and exit to happen, either it will move without utilizing energy or it will move using energy. So based on this, we see that we have active transport and we have passive transport. So what is active transport, right? So when energy is utilized for movement, right? So when energy is utilized for the movement of substances, we see that we call it as active transport. But in passive transport, we see that no energy is needed, okay? So this is one of the main point. Now in passive transport, we will explore more, right? Passive transport we have two kinds. We have diffusion and of course we have osmosis, okay? So let's understand this. So when we talk about diffusion, yes, so let's assume this is the plasma membrane of the cell. This is inside the cell, right? Now where is oxygen coming from? We know that oxygen that is there is transported by the blood and we know that it is brought to the cell. Now the thing is, outside the cell there is large amount of oxygen, but inside the cell there is only very little. And cell requires oxygen. So we see that outside, from where there is more oxygen to where there is less, right? Or we say higher concentration to lower concentration. We see that there is movement of oxygen inside, right? Now, what is happening? We see here that this movement is happening from region of higher to lower, from more to less, and there is no energy which is required for this, right? Which is why this right here is what we call as diffusion, right? So how do we define diffusion? I'll write the definition down. Very, very important. So diffusion is the free movement. When I say free movement, that means it does not require energy, right? So free movement of substances from a region of its higher concentration to a region of lower concentration, okay? Now one very important thing that you need to understand, that it doesn't matter if a semi-permeable membrane is present or no, okay? Semi-permeable membrane is there, it's not there, it doesn't matter, right? So everyone take a screenshot of this very, very quickly, right? Because diffusion is how the gaseous transport takes place. Whether it is movement of oxygen from outside to inside, or it is movement of carbon dioxide from inside to outside. Yes? Take a screenshot of this, because if I go ahead, if I change the slide, you will not be able to see this, right? Okay, very good. I will give you an example of movement, active transport. I will give you an example of cell wall, cell membrane. I'll tell you the difference between the two. I'll tell you everything. Don't worry, don't worry. I'll tell you everything. So my aim is that today by the end of it, right, it is for you to understand everything. Yes, ma'am, kindly show previous slide. Tanishka, I will write it once again with the final set of things, right? Okay. मैम कैसे बनते हैं इसमें? So diffusion में mainly they will ask you what kind of movement is it, right? Is it a passive transport or is it active transport? Then they can ask you how does gaseous exchange happen? Then it's like 1 marker or 2 marker, right? So that is something that we need to know. Active movement, I will tell you. Stroma also I will tell you. So quickly to rewind and recap this, diffusion is nothing but free movement of substances from region of higher concentration to region of lower concentration. Imagine if you take a drop of ink and you drop it in water, we see that it will spread across from region of higher to lower. So think about if you had a perfume, right? Perfume bottle mein there is more. But when you press, it will spread everywhere. The whole room will smell like that, right? So how is it that from one perfume bottle it went everywhere? Because the molecules slowly started spreading everywhere, right? Ma'am, does it happen mainly in gas? Yes, mainly with gases, and then mainly also we observe it with aerosols, right? So that is something. So are we clear with diffusion? Shall I, delete this slide? We are good to go, right? In the next half an hour by 7:30, our aim is to wind up the summary, okay? So 7:30 pe we should wind this up. Ma'am, is there daily cases? Exactly. Using of your perfume, using your, how when your mummy is cooking in the kitchen, you are able to, smell it all the way there. How is that happening? That is also diffusion, right? Hypertonic, I will be telling you. Don't worry. But I will tell you. But are we clear with this concept? Very, very important concept. Okay? What is concentration? Concentration is the amount, right? How much oxygen, how much carbon dioxide. That is what we mean by concept, Pradeep. Okay? So now let's move on to the next one. Virender, you know my Hindi is not good, so we will stick to English. Okay? So now we will move on to understanding for water. So in this, if you see, we have understood how oxygen and carbon dioxide are transported. But now what about water? Yes. Now somebody was asking me, ma'am, why can't water be transported through diffusion? Now see, one thing is for water when compared to maybe the gaseous molecules, they are slightly bit bigger. But another very important thing with water is that the constant equilibrium, okay? Equilibrium or balance must be maintained, right? Now, in the case of our body, apart from the water we drink, a lot of components are made up of water. Too much of water is also too bad, right? So, it's not that just when we drink water, water is moving. Water is also there within our body, which is why the movement needs to be regulated. Which is why in this case, we have osmosis, or we have that semi-permeable membrane that regulates this, right? So, now let's go on to the next one, right? So now, Nisha, are you clear with why diffusion doesn't work and why it is needed? Equilibrium क्या होता है? Equilibrium होता है balance. दो जगह के balance, जब balance maintain होता है तब equilibrium होता है, ठीक है? So now let's move on. Somebody is asking me, ma'am, is this chemistry class or bio class? Chemistry is there in bio, physics is there in bio, bio is there in chemistry, science, as we know, is an integrated thing, right? So now let's move on. What is osmosis, right? Yes, osmosis is a type of diffusion. I agree, okay? But we cannot say osmosis and diffusion are the same thing. Yes, that is one thing that you need to know. Now, osmosis, like how our friend Dhruv Bunny Boy has told us, we see that osmosis is the movement of water, right? So now here we see that osmosis is the movement of water molecules or solvent molecules across a semi-permeable membrane, right? So here, if they ask you for the definition and this particular part is not there, you will lose marks, right? So osmosis, yes, like how intelligent girl is telling me, osmosis is for movement of water molecules across a semi-permeable membrane, right? So here your plasma membrane acts as the semi-permeable membrane wherein we see that there is movement, right? This is still an incomplete Definition. So movement of water molecules across a semi-permeable membrane from where to where? From a region of higher concentration to a region of lower concentration, right? So everyone quickly take a screenshot of this. Quickly take a screenshot. I'm going to move aside so that you can write. Ma'am, why is diffusion? Is diffusion active or passive? This is passive again because we have not spoken about energy here, right? Ma'am, why not selectively permeable here? See, selectively permeable comes for various other components, right? And diffusion is not osmosis cannot only, it's not something that takes place only inside the cell. So if I take a potato, right, and I make a hole inside it. So in the side I'm going to draw. Imagine I take a potato, I cut it like this, where I put some amount of water here, and I take this whole potato and I put it inside one bowl with water. We see that there will be diffusion that osmosis that takes place. We see that osmosis will take place and the water level increases. So here the potato that you see on top of me acts as a semi-permeable membrane. Muslin cloth. How many of you have seen when you make paneer, right? What do? We see that the muslin cloth has fine pores through which water can go, right? So all of those things that are there is something that we observe, right? We see that also acts as a semi-permeable membrane. Yes? So what is semi-permeable? It means it allows only solvent molecules or certain molecules to pass through due to size. Yes? So are we clear? Are we clear with this? Moksha, I am telling you active transport. Ma'am, diffusion once again. See, diffusion that is there, or simple diffusion as we know, is when you have large amount of molecules, okay? And then if you see, I don't have my let's see. Yes. Now think of this sanitizer bottle, right? Sanitizer bottle ko, when I am pressing, more concentration. Now imagine this was some gas, okay? Think of this as a gas. Imagine this to be a gas, right? Now when I press it, we see that the molecules are going from this point to this point. It's getting sprayed out. Now once it gets sprayed out, it will not just stand there. It will keep wandering off to places where it can go. So it is spreading, right? So where is it going? It is again going from one space to another. It's going across the it is traveling to larger distances, right? So that is what we mean by it. Ma'am, by osmosis, not by selectively permeable membrane. Because like I told you, osmosis is not something that will take place only inside the cell. It can happen outside also, right? So are we clear? Are we clear? Ma'am, give me another example. Another example of which, Bacha Prakash, tell me. Yes? Are we clear? Have you all taken a screenshot so that I can explain the rest of it with some examples, right? Ma'am, is it plasma membrane? This here what I have drawn is plasma membrane, right? This is plasma membrane. Ma'am, diffusion takes place in alveoli also. Yes, alveoli may also it takes place, right? Somebody was asking me, ma'am, facilitated diffusion is the movement of ions, right? So normally we see that movement of ions like sodium ions, potassium ions, they would all be facilitated diffusion. Ma'am, will you be discussing NCRT or some other questions we'll be discussing? Okay. Priti, I hope now we are clear. Ma'am, diffusion ka some other examples, okay? See, diffusion ka some other examples is when you put a drop of ink, right? So if this is a dropper, right, and I put one drop of ink, we see that the ink will spread to the whole thing. This is an example of diffusion. Then next one, of course, best example I can give you is cooking. All the aroma molecules, how it will spread all the way till our room. That is also an example, okay? All right. So now we have looked at passive transport. We have looked at the two types. Now when we talk about osmosis, does it always mean that water will always go from outside to inside? Does it always mean that water molecules will go from outside to inside? Yes or no? What do you think? No, ma'am. Surbi says, ma'am, no. What about others? think? See, this is a very important topic in the chapter. That is why I am spending some time. The remaining things are very easy. Exactly. Very good. So in this case, if you see, we see that it is not always from outside to inside. It could be inside to outside as well. So what determines in which direction the water should flow? From outside the cell to inside or inside to outside the cell, right? What determines that? So that depends upon the nature of the solution or the surrounding, right? Or another very heavy term I can use is called as tonicity, right? It is not there in NCERT, which is why I am not going into the details of it. So we have tonicity. So tonicity is nothing but concentration of the solution, right? It is a concentration. Now somebody was asking me, what do we mean by dilute solution? So dilute means there is more amount of water and lesser amount of solute that is there, right? So now in this case, let's understand this properly, right? Let's go to the chemistry bit of it a little more in detail. So now how do we define? So let me just make space. So how do we define a solution, right? So we know that a solution is always a combination. So we see that it is a combination of solvent and solute, right? So when you mix solute and solvent together, it forms solution. Yes. Now we see that there are three kinds of solution, okay? So I'm going to draw three boxes. So we have three kinds of solutions. So just give me one moment as I draw. Now first kind of solution is when concentration or how much solvent you put, right? So one kind of solution is what we call as isotonic. Then you have hypotonic and then you have hypertonic. Okay. Now understand this carefully. For those of you who get confused constantly, I need 5 minutes of focus in the chat. Okay. Plasmolysis, everything I will explain to you. Don't worry. But I need you to pay attention now. Okay. Because if you understand this now, you will, I'm telling you, you will never forget. Okay. Now iso means same. Okay. Hypo means less. And hyper means more. Okay. Now I'm saying less, more, same. Now imagine if I am telling that I am taller than you, right? I am taller. But how do you know what am I taller than, right? It's not that, that doesn't mean that I am taller. I'm the tallest among them all. No, right? If I say I am taller than Anchal, that means that with respect to Anchal, I am tall. But if I compare myself with Alka, I might be smaller. So here it is with respect to something, right? So in this case, it is with respect to. So it is with respect to a certain thing. So in this case, when you think about it, imagine there is a cell which is put in this solution. Now we call this as an isotonic solution when the solvent, right? How much ever water molecules, right? And water molecules inside and outside, and if you look at all the solute also, that is also equal. Inside it is the same, outside it is the same, right? So that is what we mean by isotonic. Isotonic is with respect to each other. Okay, with respect to the cell, this solution is isotonic, right? But now if I take another cell, all right, I am taking another cell. Now when I compare this cell, what happens is that I see that outside there is lot of water, very dilute solution it is. Lots of water, okay, and very less solvent. So yellow color is solvent. Now inside if you see, water is less, okay, I mean inside the cell. And here if you see the solute is much more. So now what will happen? Outside water is more, inside water is less, right? So what will happen? water is going to move from outside to inside. So hypotonic solution is one which has less amount of solute, more amount of water, and as a result it will go from outside to inside. Yes? So if a cell gains water, normally it gains when kept in a hypotonic solution. Yes? It is kept in a hypotonic solution. Alright? So now in this case we see that cell will swell. Remember? Hypo means cell will swell. But now when I move on to another solution, right? Now here if I keep another cell, I am going to keep a, let me use maybe a bright red color. I am keeping another cell. Now in this what happens is that there is very little water, but there is lots of solute. Okay, yellow color is lots of solute. Now when compared to inside the cell may lots and lots of water is there when compared to outside. So what will happen in this case? What will happen in this case? Case number 3. We see that water will move outside because concentration of water outside is less when compared to inside. There, yes? So in this case, what do we observe? Cell will shrink, right? So in hypertonic solution, cell will shrink, okay? मैम हमें कैसे पता चलेगा कि सेल में कैसा solution है? See, if they tell you that it's a hypertonic solution and a cell is kept inside it, hypertonic solution will have more solute, okay? Hypo will have less solute. So by default, a hypotonic solution is a dilute solution. But if they tell you, if they give you a special cause, at all times, if the cell has more water than the outside, however dilute it be, the surrounding solution is hypertonic. Okay? But if cell has less water when compared to the surrounding, it is hypo. Are we clear with this? Are we all clear? Ma'am, process through which a living plant cell loses water through osmosis due to which shrinkage? Yes. Yes. So plasmolysis mein, Nisha, you can add one point that extreme shrinkage takes place. All the water will be gone out, right? So are we clear with these three? Hypo mein cell will swell. In the other one, cell will shrink. This, it will remain the same. Yes? Saroj, what confusion do you have? Tell me. This concept, if I make it clear, no? It becomes super easy for all of you, okay? Saurabh, what is cell? Bacha, cell is basic unit of life or fundamental unit of life, right? Ma'am, what is erythrocyte? Erythrocyte is red blood cell or RBC, okay? Mihir, I am coming to that, okay? Yes, all right. Ma'am, can you explain in Hindi? Kritika, my Hindi is very, very bad. You know that, right? But I will try. See, passive transport pranya examples are diffusion and your other example that is there, no? Ma'am, hypotonic mein cell will swell, but you write hypo is less. Saroj, hypo means less solute. So imagine I'm taking sugar solution, but I put lot of water and I put only half teaspoon sugar. So that means less solute. Hyper mein more solute, okay? This is with respect to the solute that is there, right? So this is with respect to solute. Are we clear? Are we all clear with this? Yes? Protoplasm, I will explain, Nisha. Please stay, focused. Bulk transport is when large amounts of, things are moved out, right? Grapes in honey, no. Raisins in water is an example of that. Raisin, dried raisin in water is an example of hypertonic solution, where inside there is no water, right? And there's so much water outside and it will go inside, okay? So now, everybody take a quick screenshot. Quick screenshot. And everyone, I hope all of you have hit the like button. I need to see at least 150 likes on this video now. See, we've been teaching for so long, so I need to see at least... Okay, I need to see at least 175 likes. Quickly. आगे बढ़िए मैम, जरूर फटाफट like कर दीजिए और we shall move ahead to the next one, right? Yes, thank you Bunny Boy for helping me out. Okay, ma'am, can we say in hypotonic and hypertonic there is osmosis? Adiba, if I put a cell or if I put a substance inside, then osmosis will take place, right? Yes. All these processes, which is movement of water molecules, take place with the help of osmosis. Yes? Very good. So now we have looked at passive transport. Whoa. Now I have active transport. Now a lot of you are asking me, ma'am, where does active transport take place? For that, let us look at our roots, right? So root of plants, we know that there are many structures here, and we know that there are things called as root hair, right? So we see that there are substances called as root hair. Now root hairs are actually just made up of one single cell, all right? And we see that root hair are mainly playing an important role in absorption of water and minerals. Now the thing is, in the case of taking up minerals, right, or maybe taking up some nutrients or taking up some ions, we see that it goes from a region of higher concentration, to a region of low concentration to higher concentration, which means that actually inside there are some essential minerals or there's good amount of solute. But on the outside we see that it's very less, but it's still there, okay? But still they need to take it up for the plant so that it can get transported. So in this case, we see that there is movement, right? From region of low concentration to high concentration, and for this it needs energy. So this right here is an example of active transport. Now in our body also, if you see, we have these sodium and potassium ions which are constantly moving in and out, right? So this sodium and potassium ions are constantly moving in and out, and at times they go from low to high and high to low to maintain equilibrium, right? Which is why in this case we call this as a active transport. So are we clear with this? Are we clear? So if we were to finally differentiate between active and passive, we see that energy is required. Right here, no energy is required. Now here we see that substances go from a region of low concentration to a high concentration. Here they go from high concentration to low concentration. Example here of course is absorption of minerals, right? Or movement of ions can also be an example, right? Movement of ions. Now at the same time, when you look at passive, you can give the example of diffusion of oxygen or movement of water through osmosis, right? So everybody take a quick screenshot of this. Ma'am, difference between osmosis and diffusion. Osmosis में semi-permeable membrane is there. Diffusion में no semi-permeable membrane. Primary difference, right? Are we all clear with this? Fatima, please take care. Please take care. Menti is the third thing. So after subjective questions only we are doing Menti. Ma'am, what is cancer in cells? Cancer in cells? No. It forms cancer cells where cell goes uncontrollable division. Take a screenshot everybody, and then I'm going to move on to the next part quickly. Yes? Take a screenshot and give me a thumbs up. Example of active transport I have written here already, Surbi. Yes. Very good everyone, very good. Yes. Now everyone, I hope all of you have subscribed to the channel, right? So all of you understand, right, that you have to subscribe to our channel because this is where all the amazing things happen, right? And all this in-depth preparation will happen only here. So everybody quickly subscribe. Tell me that, ma'am, I've already subscribed and thank you for this session, right? So subscribe because nowhere else are you going to get such quality, I would say quality explanation. Yes, very good, very good. Done, done, done. That is all what I want to hear. Now the last part in plasma membrane that we also need to understand is that plasma membrane having this kind of structure is also extremely flexible, right? So we see that plasma membrane is flexible, and we see that this flexibility of plasma membrane in some organisms like amoeba helps them capture their food, right? So we see that they're able to capture their food. Now when they engulf the food, we call this as endocytosis, right? So now in the case of amoeba, if you think about it, we know that it's a single cell organism and they will extend their false V to capture their food. So this process in which they extend and they engulf here is what we call as endocytosis, right? What is endocytosis? When engulfing happens, okay? But now once some waste materials might be there and we know that it needs to go out, right? So that is what we call as exocytosis, when from that we see that it is expelled out, right? So that is what we call as endocytosis. Are we clear with this? Are we clear? Right? So engulfing is what we call as endocytosis. When we need to release it out, right? When a vesicle that is there will have to release it out, we call it as exocytosis. Yes, very good everyone, very good. So now this was all about the plasma membrane. Now we know that plasma membrane is present across all organisms, right? But we also know that there are some plants, especially in the case of plants and in the case of fungi and in the case of bacteria, we see that they have an additional layer, right? So if this is the plasma membrane, outside the plasma membrane, they will have another one, right? So in this case, what do we call this part? Yes? Yes, Pranya. Yes, yes. What do we call? Very good. It is called as cell wall, right? So cell wall that is there is an outermost. So in these things, it is the outermost layer. Now, in the case of plants, we see that this is made up of this is made up of cellulose, right? And what is the function of the cell wall? Can you tell me quickly in the chat? Yes? Quickly in the chat, can you tell me what is the function of cell wall? Very simple and easy. Yes? Cell wall is made up of cellulose in the case of plants. And what do? They provide protection and they provide rigidity to the cell as well, right? So protection and rigidity. Now when we talk about the permeability, right? So when we talk about the permeability, we see that cell wall when compared to our cell membrane is freely permeable, right? So we see that they are freely permeable when compared to our cell membrane, which is selectively permeable. So this is what we mean by the cell wall, or how cell wall is present in the case of plants, fungi and bacteria. But one thing to know is that cell wall in fungi is made up of different structures. Cell wall in the case of bacteria is made up of different components. Cellulose is only in the case of plants. Yes, ma'am, is plasma membrane a living part? Not really. Yes, you can consider it somewhat. But no, not really. No, not really. Sorry. Okay. Am I teaching ICC board? No, this is mainly NCRT, right? Okay. Now, moving on, I had a question which I saw. Somebody was asking me, ma'am, in the case of cell wall, right? Is the cell wall flexible? Saroj, no. Cell wall is rigid. If it's providing rigidity, it is not flexible. Yes? Okay. So now moving on to the next one, which is cytoplasm. So we have covered the first component that is there, which is cell membrane. And my God, it took us good amount of time to cover cell membrane, no? Now we're going to quickly look at cytoplasm, right? So now in cytoplasm that is there, it is nothing but a jelly-like viscous fluid which is present between the plasma membrane and the nucleus, right? So we see that it is a jelly-like viscous fluid which is present between the plasma membrane and the nucleus. And we see that various chemical reactions takes place in the cytoplasm, and all the cell organelles that are there are found embedded here. Now in this case, when we talk about cytoplasm, another thing that comes into the picture is protoplasm. How is cytoplasm and protoplasm different? Now protoplasm is the total living component, right, or total living substance. Yes, substance which includes the nucleus, it includes the cell organelles and the cytoplasm, right? So all the total living components that are there are included in protoplasm. But while moving on, when we have a look at cytoplasm, it is just a jelly-like fluid. So are we clear with this? Are we clear? Alo gamer, you have to wait for Menti. Don't spam in the chat. Yes? Okay. Ria, I'll not go into cytosol and what is trophoplasm because it is a little beyond what we are doing for NCRT, which is why I am not going to move into that. Okay? So now with this, we are going to move on to cell organelles. Okay, we are going to look at all the cell organelles that are there, starting with the most important one, which is the nucleus, right? So how do we define nucleus at all times, right? How do we define nucleus? Now the nucleus that is there is the control center, right? So we know that it is the control center of the body. Now in the case of an animal cell, right? So if I were to take the example of an animal cell, we see that the nucleus is double membraned, right? So we see that it is double membrane, right? Or you can say that it is double layered, and this is called as the nuclear membrane. And it's not a continuous membrane, right? We see that there are some pores which are present, which we call as the nuclear pores. Yes? And inside this also we find some jelly-like fluid here, which is called as the nucleoplasm. So the jelly-like fluid inside the nucleus is called as nucleoplasm. And we also see that there is a structure called as the nucleolus, yes, which has a lot of which also plays a key role in protein and ribosome synthesis, right? Now on the other hand, we also see that there are structures present within, which is the genetic material, right? Now what is the genetic material of our body, right? What is the genetic material? Can you tell me? Very quickly everyone, what is the genetic material? DNA. Yes, Tanishka, that is enough. Viscous means jelly-like. Okay, very good. The genetic material is DNA. What is DNA? DNA is deoxyribonucleic acid, right? So that is what we mean by DNA. Now this deoxyribonucleic acid is what consists of all the information. So if I am saying that the nucleus is the control center, it is what is responsible for regulating various activities that happen within the cell. Who is actually doing it? It is the DNA, right? And we see that the DNA is like a helical structure here, long structure. And we see that there are, it has all the information. Now the DNA is very long. If you take the DNA out of the cell, it's almost 2 meters long. But you imagine something which is almost 2 meters long is found condensed inside our body. It's found condensed inside a microscopic cell. So how exactly that is possible? So we see that there are some DNA and proteins which are condensed, right? So we see that they are condensed. So think of if you had a long rope and you had to fit it inside your bag. What will you do? You will take that rope and then you will make it into round, and then you will make it smaller and you will fit it inside, right? So that is what DNA and proteins together, they will be condensed, or they will be made small, not smaller, but more compact, right? So they are made more compact and they form chromatin material, right? So what is chromatin material? It is thread-like structures, okay? They are thread-like structures. Now, this chromatin material, when it will become more condensed, all this thread you take and make it more condensed and it will become into a rod-like structure called as chromosomes, right? So, chromosomes are rod-like structures which usually arise especially during cell division time. So, we see that these chromosomes at the end of the day are made up of chromatin material. मंगलम सुरभि इंस्टाग्राम यूज नहीं करती है, आप क्लास पे फोकस कर लीजिए, ठीक है? What is DNA made up of? DNA is made up of deoxyribose sugar. It is made up of, phosphate group, and it is made up of many other structures, right? So Meenu, I hope I have cleared your doubt. Ma'am, is DNA present in each and every living cell has DNA in it, okay? Every living cell has DNA, all right? Okay. Yes, all right. Nucleic acid, yes. So are we clear with this? Are we clear so far? Give me a quick thumbs up. Give me a thumbs up everyone. Are we clear? Chromosomes क्या होता है? What is chromosome? Chromosome is a rod-like structure which is made up of DNA, right? It is made up of DNA. Yes. Meiosis, my tosses, I will tell you. Don't worry. Okay. Now everybody take a screenshot because now again when I do this it will go. Okay, I'll erase it. RNA, I am coming to the point. Don't worry. Don't worry. Take a screenshot and I'm going to remove this. Thank you, Satyam. Thank you so much. Plasmolysis, Kritika. Yes, I will explain that. I think I missed it a little earlier. Okay, very good. All right. So see, one thing that you need to understand, right, is that the chromosomes that I was telling you about, chromosomes are the ones which play a key role, right, or they play a central role in cellular reproduction, okay? So the chromosomes are structures which will form during cellular reproduction, okay? So during cellular reproduction, we see that chromosomes are formed. Now, another thing all of you are asking me is, ma'am, what is RNA? RNA is ribonucleic acid, all right? So this is ribonucleic acid. Now, if you look at DNA, DNA is our genetic material and controls all the functioning. While RNA that is there is mainly responsible for protein synthesis, right? So this is responsible for protein synthesis and carries out various other functioning, right? So this is the main role. Are we clear with this? Yes. Reproduction in humans is in 10th standard. You're still in 9th. Okay? All right. So now moving on to the next part. So this is all about nucleus. Now, if you remember, I told you based on type of nucleus, we have two kinds of organisms, right? So, based on type of nucleus, we see that we have two kinds of organisms. We have prokaryotic organisms and we have eukaryotic organisms. Yes? So, prokaryotic or eukaryotic organisms are those that we just discussed. So, eukaryotic organisms are those which have a well-defined nucleus, right? And we see that the genetic material is enclosed in a double membrane, or it is enclosed in a membrane bound structure. Okay? So we see that plant cell, animal cell, they are all examples of the same. But moving on the other side, right? In the eukaryotic cell, prokaryotic cell, we see that no well-defined nucleus, right? And we see that the genetic material is found concentrated in a particular area, right? So it is found concentrated in a particular area. And we see that this particular area is what we call as a nucleoid region, right? So this is what we call as a nucleoid region. So everyone, pakka 2-mark question will come on this. Differentiate between prokaryotic and eukaryotic. So everybody quickly take a screenshot. Harsh, I have already explained DNA, RNA, rewind and you can watch it. It is there. Ma'am, genes क्या होता है? Genes are fragments of DNA, right? Or they are sequences of DNA that code for a particular character, right? Okay. Ma'am, difference between nucleus and nucleoid. Nucleus is the structure, double membrane structure which has the genetic material. Nucleoid is a region, okay? Understand. One is a structure, another one is a region, an area, all right? Okay. Are we clear with this? Are we clear? Are we good to go? Have you all taken a screenshot? Yes, example here is bacterial cell, right? Bacterial cell is an example of this. Yes, if you're a JEE aspirant and you're interested in biology, I'm very happy. I have a very interesting question. Ma'am, does this mean that eukaryotic cells cannot transfer their genes or similarities to their offsprings due to... No! Who said? Genetic material is there. Eukaryotic cells pay most often than not. In plant and animal cells, their genetic material is DNA and it gets transferred from parent to offspring, right? So that is definitely there. Kritika, is it okay if I explain plasmolysis once I finish the whole part? I'll finish all the cell organelles and then I will come here. Yes, all right, okay. Prokaryotic, eukaryotic is not clear. See, eukaryotic cells are your plant and animal cells, right? So we see that here, if you see, when there is a nucleus that looks like this, genetic material inside the nucleus, it is eukaryotic. Prokaryotic is when there is only in one region it is found, all right? Very simple and easy, okay? Now moving on to the next one. I'm going to get rid of this so that you can see. Now the next cell organelle is mitochondria. Now mitochondria is most often than not known as the powerhouse of the cell, right? And we know that this mitochondria is necessary for the production of energy, right? And we know that energy is produced in the form of ATP. Now mitochondria that is there is a double membranous structure. It is made up of two membranes, right? And we see that the inner membranes are folded into finger-like projections to manufacture or to produce more ATP. And along with that, we see that they have their own ribosomes, which are another kind of cell organelles which we will learn now, which is necessary for the production. So this is another type of cell organelle which plays a key role in protein synthesis, right? So we see that ribosomes play a key role in protein synthesis. And if ribosomes are present inside the mitochondria, we see that they produce their own proteins, right? Very simple and easy. Now moving on to endoplasmic reticulum. So Shiva, I will give you a break once I finish chapter summary. I'll give you a break after that. Yes? So now we have endoplasmic reticulum. Now when we talk about endoplasmic reticulum, we see that they are these flattened tubular structures, right? And we see that these endoplasmic reticulum that is there has two kinds, right? So if you talk about the general function of ER, so what is the function of ER? ER plays a key role, or they serve as channels to transport material, and they also act as a cytoplasmic framework, right? So they provide a framework for various chemical activities to take place. Now when we talk about this, right, we have two kinds. So in this case, we have two types of ER. So in the chat, can you tell me? Yes, in the chat, what are the two types of ER? Ma'am, what is Krebs cycle? Can you explain? See, production of energy takes place in three steps: glycolysis, Krebs cycle, and electron transport chain. You will learn anyway in detail in 11th standard, and then you will get very scared looking at it, but there's nothing to be scared about. So it's a cyclic process wherein we see that some amount of ATP gets produced, and we see that it also results in the production of some hydrogen ions which are needed, right? So that is what we mean by it. Okay. Now moving on, yes, like how all of you are telling me, we have SCR and RER. So what is RER? It means rough endoplasmic reticulum. And why is it rough? It means that on the surface, they have ribosomes, right? Giving it a rough structure. Now, if ribosomes are present on the surface, what is the function? Yes, what is the function of RER? Active transport, bacha, I already told you. It is transport of minerals, transport of ions. They are examples. Menti will happen in another 1 hour. Harsh, hashtag we want break. Hashtag 8 o'clock you will get break. Very good. So this is synthesis of proteins, right? So synthesis of proteins. So whenever ribosomes are there, tell yourself, right? Tell yourself that it is protein synthesis. Now moving on, we have SER. SER here stands for smooth ER or smooth endoplasmic reticulum. That means that there is no ribosomes. Now what is the main function of this? The main function of these particles that are there, right? The function of the smooth endoplasmic reticulum is for the synthesis of lipids, right? So we see that they mainly play a key role in the synthesis of lipids. Now we also see that in some cases, right, especially in liver cells, right, in the case of liver cells, we also play that they play a key role in detoxification, right? So they play a key role in detoxification and getting rid of maybe unwanted toxins that are there. So these are in liver cells of vertebrates, right? So that is something that you need to remember about RER and SER. Take a screenshot, everyone. We are done with ER. We'll go to Golgi apparatus. Yes? Take a screenshot. No, Harsh. I'll finish it soon. See, I don't mean it to hurt anybody. I hope all of you all are not getting hurt by what I'm saying, right? It's just that at times it takes a lot of time to stay focused. So Harsh, please don't take it personally, bacha. I don't mean it, right? Yes, exactly. This lipid synthesis and protein synthesis, at the end of the day, we need it, right? We know that it is required for membrane biogenesis. What is membrane biogenesis? Synthesis of membranes, right? Okay, very good. Take a screenshot and we are going to move on to Golgi. Swedberg unit, RT, is a unit of how we measure the particle, right? So in ribosomes, if you see, we have 80S, 20S and 70S. So the S that is there is Swedberg unit that is there. Okay. Yes, Manu. Yes. Okay. Clear, ma'am. Okay. All right. DDT is it's some pesticide that is there. I'm explaining Golgi, I'll explain vacuole, I'll explain everything, but I hope you've taken the screenshot because after I change it will not come back. Yes. Very good, everyone. Very good. So I think the next one is ribosomes, right? What do ribosomes contain? See, ribosomes are made up of structures. So a ribosome will look something like this, okay? Which is why it will have 2 units. And ribosomes, as we know, that are there, are responsible for protein synthesis, right? So they are necessary for protein synthesis. Now moving on to the next one, which is Golgi apparatus, which is what everybody looks like they're scared of. I don't know why. Golgi is very, very easy, right? So who discovered Golgi, or why did the name Golgi apparatus come into the picture? How many of you can tell me where did the name Golgi come from? Yes? Where did the name Golgi come from? Very good. Camillo Golgi. Yes, Camillo Golgi was the one who discovered this, right? Now when you talk about Golgi apparatus, now we know that the RER is responsible for synthesizing proteins, right? Because they have ribosomes. Now the thing is, these proteins may not be fully functional, okay? So they are not fully functional, right? So you think about it. Imagine a car just came out of, production, okay? From the manufacturing industry, the car has just come. But the parts need to be assembled. Then the engine needs to be seen if it is working properly. We need to paint it on the outside. So we need to do all of this, right? So in this case, what happens? There will be other people who will put all these things together, or they will be packaging it, right? So they will be packaging it. So that is the job of Golgi, right? So what is Golgi? Golgi is also known as the postman, right? Golgi is known as the postman of the cell. Study point, please don't spam saying I'm ignoring you. I am not ignoring you. You didn't send me your doubt. That is why, right? So what is the function of Golgi? Function is packaging, right? It is packaging, transport, or I will say modification. I want to write it in order. So it is modification, packaging, and transport, right? This is the function of it. Now when you talk about Golgi, right? We see that they are made up of many flattened structures, okay? So there are many flattened tubular structures which are called as cisterns, okay? This is there in your NCERT before you come and fight with me saying, ma'am, it is not there. So cisterns are what? They are flattened tubular structures, okay? So they are flattened tubular structures that make it. Now in this case, we see that there is one part here, right? So Golgi will look something like this, okay? Now there are 2 parts. Now one part here is the cis part and the other one is the trans part. Now the cis part is the part which receives the vesicles which are nothing but small vehicles, okay? Think of it as small cabs which the protein takes to reach it, right? So we see that the vesicles will reach Golgi. Now here inside this, we see that it will undergo modification. And finally, we see that they will be ready to be transported through secretory vesicles, right? So we see that secretory vesicles that will be there will transport it. So cis is the receiving part of it, right? And we see that it will undergo modification, and after that at the end of it we see that the trans part is where through which the transport takes place post modification, right? So once modification has happened inside the Golgi, it will reach the trans side of Golgi and bye bye, time to go, right? So that is something. Again, who is this? Nikhil Kumar asking Garima for Instagram. Nobody is asking anybody for Instagram here. Next 3 years, nobody is talking about Instagram. Okay? Ma'am, ER and transport and Golgi also transport. If the question comes which is mainly response for transportation, which organelle will you write? Very interesting question. If that is the case, which organ? They'll tell you very clearly. Which organ is responsible for transport of modified proteins or are responsible for transporting proteins from inside to outside, right? So they'll catch all this. I mean, they'll be specified. So if it is specific, then you can write Golgi apparatus primarily, right? You have to write Golgi as the answer. Okay. Nikhil Kumar, stay focused in class. Otherwise, you know, moderator will block you. Yes, okay. Exactly. Very good. Kriti has told me one very important point, right? Okay. So now moving on, another thing which Golgi is also responsible is for the production, right? So when we talk about Golgi, we know that it is also responsible for the formation of lysosomes, right? So lysosomes is also something wherein the formation takes place. So the production of enzymes, the enzymes which are present inside the lysosomes happen in the RER. And when they come here, they'll undergo modification and they will go. Yes. Are we clear? Are we clear? Niksha is like, ma'am, difference between... Yes. Okay. Are we clear with Golgi? All of you who had doubts with Golgi. Niksha, I will take your doubt with difference between Golgi and dictyosomes. I'll get it clear. Yes. Lysosomes I will explain. Right? My aim is, I told you, 8 o'clock I have to give you a break. Because then we will finish with this. Right? Bacha, pro legend, please go back and check for cell wall. I've already explained. Yes. Very good. So now, have you all taken screenshot? Because I'm going to, this will go. This will vanish. This will vanish. So last time I'm telling you. Ma'am, structure of Golgi, they are made up of flattened structures here as you see, which are called as cisterns. We see that there is a cis-facing side and a trans-facing side. Cis gets all the proteins and we see that modification takes place across and through the trans side it will go, right? Yes, all right. After taking, give me a thumbs up so that I'll come on screen. Very good, very good. Okay. Now going on to the next one, right? So somebody was asking me what is the function of nucleolus. It is for production of ribosomes. They are responsible for production and assembling ribosomes, right? So now the next one somebody was asking me was, ma'am, Golgi and dictyosomes, right? See, Golgi, as we know, are made up of all these flattened structures. But dictyosomes are structures which are found in and around Golgi, which are slightly scattered, okay? They are slightly scattered like this. So if this is Golgi, no? In and around it, you will find it scattered. And this structure is what we call as a dictyosome. And this is something that we find in plant cells, okay? So that is something about it. Now we see that Golgi apparatus that is there. Arpit, I have already explained RNA, rewind and you can watch back, okay? So now of course, as we know, next up that we have is lysosomes. And lysosomes are formed or produced by the algae, right? So next up is lysosomes, yes? Now what are lysosomes most oftenly known as? Tubules means tube-like arti, tube-like structures, okay? What do lysosomes also know as? Ma'am, function of dictyosome, same function as your Golgi. Just that there are additional structures which help us, right? Yes, okay. Not social bag, suicide bag. Yes? Lysosomes are not social people who like to interact and go out, but they are also commonly known as suicide bags of the cell, right? Now, why are we calling them as suicide bags? So now when you look or somebody's telling waste bag. That's actually very nice, but yeah, suicide bags, okay. Okay, a 3, sorry about that. Yes, all right. So now, of course, when we talk about lysosomes, right, we see that lysosomes that are there are membrane bound. They're like sacs, okay, sac-like structures. Now, inside the lysosomes, there are many digestive enzymes, all right. So, they are digestive enzymes, or what we also call as hydrolytic enzymes, okay. So, we call them as hydrolytic enzymes. Now, why are these lysosomes necessary? Now, the thing is, it's not that the cell is always perfect, right? So, cell is not always perfect. But as a matter of fact, sometimes there will be some worn out part of the body, right? So, there will be some worn out organelles. So, worn out organelles might be there. Sometimes, you know, there might be some disturbance within the cell that is there. As a result, the cell might not function properly. So, we see that when all these worn out or worn out parts are there, or if at all some foreign substances, right? So, maybe some dust particles or maybe some foreign organism enters into it. In all these cases, this is disturbing the balance of the cell. So what will this lysosome do? It will immediately go and it will engulf, right? So we've learned about endocytosis already. So we see that it will engulf it, take it within, it will destroy it within itself, right? So that is something that we need to do. Now we also see that at times, we see that if the cell is not functioning properly, the lysosome will make sure that the enzymes are released into the cell and they will destroy the cell, right? So why does the lysosome destroy the cell? Most often than not, if the cell only gets damaged, right? So if the cell gets damaged, then we see that there's no use of the cell anymore. We need to get rid of it. So at that time, we see that the lysosome will explode, right? So they will explode or they will burst open, releasing the enzyme, right? Yes. Ma'am, why is it that when a virus enters the cell, they are not able to destroy? See, the thing is, there are various ways in which they will detect it, right? So they will detect the presence of the virus or no. At times, if the lysosome is not capable of it, what happens is that in the case of virus, right? If the virus comes and it sits here, it will only inject its genetic material inside, such that the genetic material will go inside the nucleus and they will bind with the genetic material of our cell and use that to their benefit. So it's not really helpful. And yes, it's most often then known as scavenger or housekeeper. Ma'am, where lysosome goes after destroying the cell? After destroying, it is also gone. Tata, bye-bye. That's why suicide, gone. It's over. Ma'am, during cancer, see, during cancer also, what happens is that the mechanism in which, at times what happens is cancer cells will override the mechanism of the lysosome. That's why they'll continue to grow and to form tumor, right? Yes. Ma'am, what is hydrolytic enzyme? Hydrolytic enzyme is a way in which the enzyme acts, okay? So basically what they will do is they will cut the, like wherever there is a bond, there's a specific bond, it will go and it will cut there, right? Yes, okay. So are we clear with what is hydrolytic enzyme? Why is endocytosis found only in animals? Endocytosis is found only in animals because you know in plants they have cell wall. Cell wall is rigid, right? So when cell wall is there, they are not able to do it. And we see the lysosomes are not present that many in number in the case of plants when compared to animals, right? Which is why most often than not we find that. So everybody go ahead and take a screenshot. Take a screenshot quickly. Okay, very good. Ma'am, enzymes present in lysosomes. See, I don't have a specific name, but normally they are called as proteases, right? So we see that they are proteases which have the ability to break down proteins. Like that they'll have like, you know, various other enzymes of that way. तुमसे बात करना है, Nikhil, stay focused in class. I'm going to catch you, otherwise our moderator will block you. Okay? 6 more seconds. All right? 6 more seconds it seems. 6 more minutes. And then of course after that we are going to take a break. Are they double membraned? Yes, they are. Ma'am, what all organelles are double membraned in our body? Most cell organelles are double membrane except for the ribosome, which is single membrane, right? Which stain, bacha, I am not sure. SK shots, I am not sure of which color. I'll get back to you on this, okay? Now quickly moving on to next cell organelle, which is plastids, right? So we are just left with plastids and vacuoles, all right? So now when you talk about plastids, yes. So when we talk about plastids that are there, no, we see that plastids are basically structures which we find in plants. Yes. So what are plastids? Can you tell me very quickly in the chat, right? What are plastids? Plastids are structures, or they are cell organelles which are found only in plants. But what is the role of plastid? Yes, very good. Ma'am, main function, Kriti, I have already explained, right? I've already explained this. Very good. Ma'am, how does amoeba obtain its food? Simply, I've already explained. You can go back and rewind and check that out. Okay. Gives color. Very good. So, we see that plastids have certain pigments that are there, right? So, now in the case of plastids, we see that there are three kinds of it, right? So, we have chromoplast. We have chloroplast, and then of course we have leucoplast, right? So chromoplasts that are there, they have certain colored pigments in them, right? So we have maybe some examples of pigments are xanthophyll, carotenoids, all of them are examples. Yes. Now in this case, right, when you talk about chromoplasts, we see that we mainly find them in petals, we find them in fruit peels. Wherever we find color, we find them there. Now, chloroplast, you can say, is a kind of chloroplast which has chlorophyll in it, right? And now, in this case, we see that they are mainly found in the leaves, which imparts green color. Now, when you look at the structure of chloroplast, we see that this is also a double membrane structure. And inside, we see that there are these sac-like structures, okay? Stack-like, disc-like structures, which are called as grana. So this whole thing is called as a grana. And we see that there is some empty space around it which we call as stroma. So somebody who was asking me, ma'am, stroma and grana, what is it? So grana is the stack, stroma is the space inside the chloroplast, for those of you who are asking me. And each one thing that is here is called as a thylakoid, okay? It is called as a thylakoid, and this chlorophyll is present in this. Now moving on to leucoplast, right? What is leucoplast? They are colorless per se. They don't have any pigment. But we see that they store important substances such as starch, oil, proteins, and all of it, right? So that is what we mean by it. So what do we mean by chloroplast? So again, plastids are cell organelles which we find in plants. Yes, chromoplast, which is a colored pigment where we find in the case of petal and peel, right? And then chloroplast is chlorophyll found in leaves. Leucoplast in starch and oil. You can take a screenshot. Ma'am, difference between plastid diagram and mitochondria. See, this is chloroplast ka diagram. Mitochondria, when you will draw like this. Okay? So this is your mitochondria. Are we clear? Ma'am, who takes the place after getting who takes the place of the cell after getting destroyed? A new cell will come in its place. Okay? Yes, very good. आप सर इंग्लिश, मुझे लगा आप सर इंग्लिश बोल रहे हो, सिर्फ इंग्लिश बोल रहे हो. Oh, no, I know little Hindi, but not very good Hindi. That's the only difference. All right, what is granum? See, single one is called as granum. One single stack is granum. All stacked together is grana. Okay? Light and dark reaction you will learn next year. So don't worry about it now. So now with this we will move on to the last cell organelle, which is vacuole, right? So vacuole that is there, right? So vacuole are nothing but the storage sacs, right? So what is their role? They store a lot of important substances, right? So when we talk about what they store maybe, proteins, sugars, right? And we also see that they store some amount of amino acids. and at times they also store some excess water. Now in the case of plants, right, in the case of plants, they have bigger vacuoles, and we see that some amount of water is also being stored in it. And in the case of amoeba, we see that these vacuoles that are there are used in the case, or they are gotten rid of, right? F0, F1 is only there, F0, F1 is there in this one, mitochondria, okay? Yes. All right. So with this, of course, we come to the end of learning about all of this, right? We've learned about all of this. Now the last part, of course, is cell division. Now constantly we know that all cells arise from pre-existing cells, right? So how is it that they are arriving from pre-existing cells? By the process of cell division. So we see that new cells are formed in order to replace old, dead, or worn out cells, right? So that is why we require cell division. And how do we define cell division? So cell division is the process by which new cells are made, right? So that is how we define cell division. Now at the same time, we see that we have two kinds, okay? We have mitosis and we have meiosis. Now you don't have this in detail, you just need to know basic. So mitosis is a type of cell division which takes place in most of the cells in our body. So most cells in our body divide through mitosis, right? So we see that here if there is a parent cell, right? So if this is a parent cell and we see that it has, these many chromosomes, right? So normally in our body we have 46 chromosomes. So if 46 chromosomes are present, we call it as a diploid condition, because this 46 is actually 23 pairs. So both the pairs of chromosomes are there, so we call it as diploid, right? So this is what we mean by diploid. Now in this case, what happens is that this parent cell will undergo division in such a way that the daughter cells are the same, right? The daughter cells will also have 46 and 46 chromosomes. So a diploid parent will give rise to diploid daughter cells. So this right here is what we call as equational division. Equal parent and offspring, or parent and daughter cell have equal number, right? Which is why we call it as diploid. Diploid means 2n or 2 pairs of chromosome. But on the other hand, when I talk about meiosis, right? In the case of meiosis, we see that this happens in the reproductive cells or in the gametes, right? And this mainly takes place in the reproductive organs, where we see that there is a parent cell, right? So we see that there will be a parent cell with 46 chromosomes, right? But we see that after divisions, I think the spelling is wrong. But we see that after multiple rounds of division, the daughter cells or the gametes which are produced will have only 23. So it has half the number of chromosomes. From 46, it has become 23. So this condition where only half the number of chromosomes are present, we call it as haploid. And here there is a reduction in the number of chromosomes. So we call it as reductional division, right? So we call this as reductional division. So main point you need to know is in mitosis you will have same number, while in meiosis it will become half, right? Okay. So with this, of course, everybody, we have completed our chapter summary. It took us at least 1 hour 45 minutes, but now the questions will become easy. I will not spend too much time in question, right? So these questions we will go fast, fatafat. Germ cells are nothing but gametes, okay? Now I'll take the rest of the doubts. So I'm going to give you all how many minute break do you want? Shall we all meet at 8:10? So break time of about 7 minutes, right? We will resume at 8:10, okay? No, see, take a break because it's good for all of you only, right? So 8:10 we will resume and we will wind this chapter up at least by 9:30, okay? It's okay, you will think you don't want a break, but it will be good, all right? So everybody, break time, I'll be back. Are we ready to go? Welcome back, everybody. Welcome back. How are you guys feeling after your break? Are you feeling good? Are you feeling relaxed? I could see that all of you were talking to each other. I was reading your chat also. You all know that, right? I read your chat at all times. Yes, finally. Yes, welcome back. I can see many of you went away and you didn't come back only. Our live audience went back a little more. It's gone down. But yes, see, the reason why I spend some time with the concept is because with the questions, then you will be able to answer very easily, right? It's going to be very simple and easy. So first, our aim is to do 15. So our first milestone is done. Chapter summary. Menti kab hoga? In another half an hour, after which we wind up our 15 subjective questions. Now the thing is that now that you know the concept, the rest of it becomes very easy, right? So everybody, those of you who have still not liked this video, we need to touch at least 250 likes on this video before I end the live stream, right? So quickly go and like this video, right? Do not forget to subscribe. And let me tell you, if you feel like, Arey ma'am, why should I sit 3-4 hours in your class, right? Why should I spend so much time? It's very important that if you just dedicate these 2-3 hours, which I know is not easy, by the way. I mean, it's very simple, easier said than done for me, right? To tell you that, bacha, pay attention in class. But I'm telling you that if you pay a little attention, you give this time, all your preparation for this chapter is done. Then you don't need to come back and revisit this chapter. Which is why I tell you, if you feel like, ma'am, mujhse nahi ho raha hai, I'm feeling ki I'm getting a little demotivated, I'm feeling like I can't do this, then in those cases you have to tell me that you're feeling this, because I will push you back up, okay? I will tell you that it is possible, and I know it is possible. Yes? So are we ready? Are we full of josh? Shall we put it on fire and start solving the questions? Ma'am is not ignoring you, Bhavya. I am seeing your comments only. Yes. Ma'am, which books to practice? Start with NCRT exercise, then go to exemplar, then go to any other reference. Okay? Anshika for your this one. So let's get started, everybody. First, we're going to have a look at the 15 subjective questions, starting with the first one. Okay? So the first question is very simple. What are the consequences of the following conditions? So this is going to be a 3-mark question that you can get, right? So the first part is a cell having higher water concentration than surrounding medium, right? Ma'am, ye pakka aayenge. Haan, this is on isotonic, hypertonic. So see, they're not asking what solution. They're asking what is going to happen. So if a cell has more concentration than the surrounding medium, what will happen? Very good. Who gave me the answer like that? Anshika Gupta, well done. Water will go out, right? So that means cell will shrink in this case. Yes? So cell is going to shrink. Second one, cell having low water concentration than surrounding medium. Then we know that if this has low water concentration and this has high water concentration, this is a hypotonic solution. So cell will swell, right? So this is cell will swell. And if cell and what surrounding medium has equal, what is going to happen? Yes, cell will swell for B. What is going to happen for C? Quickly tell me. C may what will happen? Nothing will happen, right? So nothing is going to happen because we know that it is an isotonic solution. So here's a quick reminder. In hypertonic solution, when the water is more inside the cell than outside, cell will shrink, right? Now when kept in a hypotonic solution, cell will swell. And what about the third one? If it is the same concentration, there will be no change. So when you're writing the answer, you will have to structure it this way, right? Now, of course, the way it is written is very technical, but write it in words that a cell which has higher water concentration than the surrounding medium, water will move from inside the cell to outside, and as a result, it will shrink, okay? You need to write it in that way. Only then will you get complete marks. Otherwise, you'll be like, ma'am, my bio teacher is just chumma cutting marks. I don't know what is the reason behind it. That is because you need to mention both the points. That water concentration is different. It is moving outside and that is why it is shrinking. Okay. You can't just simply write and tell, ma'am, cell shrunk, that's it. No. Only half mark you will get. Only if you answer this way, you will get 3 marks. Alright. Very important. Now moving on to the next one. Draw a plant cell and label the parts which do all of this. Now this question can easily come for maybe 4 marks, where 2 marks you will get for diagram and 2 marks for labeling. Okay, half mark for labeling and identifying, or at times this can come even for 5 marks. So first thing I want you to tell me is what is what here, right? It determines the function and development of the cell, right? See, Aditi is like, ma'am, ye question aaya tha. Exactly. These are very important questions that we've picked up. Very, very important one. Yes. What is part A that determines the function and overall development? Yes, it is the nucleus. Second part. It provides resistance to microbes and it withstands hypotonic external mediums without injury. Very, very fancy question, right? What is the second one? What is part B here? Yes, what is part B? Provides resistance to microbes and withstands hypotonic external mediums without injury. Yes? Yes, I'll give you time to answer. Cell membrane or this one? What is it? Sorry, my bad. Yes, I showed you half the answer, but it is cell wall, right? So when we talk about it, we know that when water is exerted or when pressure is exerted on it, we see that there is pressure that gets exerted outside, right? So whatever pressure is exerted on it, it will exert pressure backwards, maintaining it, right? So that is what we call as wall pressure. So this is cell wall. Packages materials coming from endoplasmic reticulum. What is packaging materials coming from endoplasmic reticulum? Why not cell membrane? Because see, cell membrane is flexible, right? It is not providing any resistance to microbes per se, right? This especially external medium, because what happens is that when cell membrane is there, it will expand due to its flexibility. It's not withstanding. It will keep expanding and after a point it can explode, right? So that is why it is not cell membrane. Answer is Golgi apparatus. Yes. You guys are giving me some external answers also. Golgi apparatus. It is a fluid containing inside the nucleus. What is the fluid inside the nucleus? How will alcohol pass into plasma membrane? C2H5OH is ethanol, right? Ethanol will not pass inside. Yes. Nucleoplasm. Very good. This is nucleoplasm. Last one. It is the site of biochemical reactions. Site of biochemical reactions. I told you there is a place which is necessary, right? Lot of important substances or lot of important reactions take place here. What is it? Very good. It is cytoplasm. So now here you will get 1.5 marks, right? So if I do 2.5 marks here, you will get just for identifying this. And the remaining 2.5 marks will be for drawing this, right? So how many of you know how to draw plant cell, or do you want me to type, right? Do you want me to draw and show it to you? You're comfortable drawing plant cell, right? All of you are comfortable drawing plant cell. Ankita ma'am has already taught you, no? Or how many of you want me to draw? If you say me, then I'll not know. Tell me that, ma'am, please draw. You can tell me that. Okay, how many ion channels? There are many. There's calcium ion channel, there's potassium, sodium ion channel. These are a few which are at the top of my mind. No, ma'am, please draw. I am getting mixed this one. So I'll anyway quickly draw it, right? Then don't tell ma'am you took extra time. But see, first one when you are drawing, so you can draw a mesophyll-like cell, right? You can draw one mesophyll-like cell. Then of course you need to draw the cell membrane, okay? So you will have to draw the cell membrane. Now after this, what you need to then draw is the nucleus, right? And we know that especially in the plant cell and everything, the nucleus is present towards the side. And this is the double membrane nucleus. Draw this and then draw the... Then of course you can draw the genetic material, right? So this is the thread-like chromatin that is there. Now after this you have to draw, when you are drawing you should draw everything, right? So from here you can draw the endoplasmic reticulum. Okay, this I am drawing very roughly. So one side you can keep smooth, one side you can keep rough, right? And draw the parts which are mainly necessary for labeling, right? So then after that you can draw the mitochondria. But before you draw the mitochondria, we know that inside this there are large vacuoles, right? So you can draw a large vacuole. Then after that you can draw the mitochondria. So I'm going to use this color. So you can draw mitochondria, right? A few mitochondria here. I'll draw 2. Then of course we know that Golgi per se we will not find, and lysosomes you don't find that much. So you can draw these parts, and then you can draw white dots here to indicate the cytoplasm, right? So these are a few ways, simple ways in which you can draw. And after this you can label part by part. So the first one that was there that we labeled was the nucleus, right? So then you label nucleus. Then of course you label nucleoplasm. Then cell wall, and then of course what did we label? We also had cytoplasm. And what was the other thing I forgot? What was our other one? That was Golgi apparatus. I forgot to draw Golgi. Yes. So in this case, my bad that I didn't draw. I will use red color and I will draw Golgi here, right? Okay. Yes, ma'am, what are dictyosomes? I have already explained this, right? So you will have Golgi and then you can label Golgi apparatus. Very simple and easy, right? Dictyosomes are these structures which spread out from the Golgi, right? That is what it is there. Lysosomes, chottu sa, you can draw 1 or 2, but very few, right? Ma'am, peroxisomes are present in plant. Yes, we observe it, right? Okay, very simple, easy diagram that you can draw. You can take a quick screenshot, everyone. Quick screenshot. Now for those of you, I know I missed explaining plasmolysis. Now plasmolysis is a kind of shrinkage or exosmosis that we observe in the case of plants, where we see that when all the water moves out of the plant cell, it will shrink, right? So it will shrink altogether, wherein if this is the plant, if this is the cell wall, then we see that this is how much it will be there, leaving a lot of space between it. So a condition where all the water has removed or most of the water has come out and the protoplasm, the plant cell has shrunk to a point wherein only protoplasm is there, we call it as we call this as plasmolysis. All right. Have you all taken a screenshot? Yes? Ma'am, you forgot to write. Okay, cell membrane. Actually, I only labeled the parts which were needed for the question, right? Harshit Yadav, I am not ignoring you. Menti is only at 8:45. Till then, nobody should ask me about Menti. Let's keep 8:50 as our target to finish all of our questions, okay? Yes, all right. So you will get 5 marks for drawing this. Now moving on to the next one. Very, very important question, most likely to get 3 marker. Difference between plasma membrane and cell wall. Now you only have to tell me this. We have already discussed. So you have plasma membrane and cell wall, right? All these answers you should only give me. We've already discussed this. Quickly tell me, Harshit, why? Why are you feeling like I'm ignoring you? Ye exam mein aaya tha. Exactly. Very good. Cell walls are dead. Okay. Meenakshi, why all of you are telling I'm ignoring you? I'm not ignoring you. Okay. Yes. Outermost. Okay, very good. So plasma membrane, can I say, is found both in animals and plants. And this is found only in plants. Fungi, and in some bacteria cells, right? Yes, very good. So some of you are telling me, ma'am, plasma membrane is made up of phospholipid bilayer. Very good. And this is made up of what? If you're talking about in plants, right? In plants, we know that it is made up of cellulose. What else? Plasma membrane provides protection. Okay, it does provide protection. Cellulose, very good. Cellulose is there. I'll repeat the question, SK Dow, don't worry. And happy birthday, Abdul. Happy birthday. Hope you had a wonderful day. Yes, what else? Provides protection. Surbi, very good. That is what I was looking for. This is selectively permeable. Yes, while this is freely permeable, right? So this right here is freely permeable. Now what else? That means this will allow selective substances to pass through. This will allow all substances to pass through, right? Now at the same time, when we talk about this, we know that this the main function, right? So that is what we are still looking for. So function is, of course, we see that it provides flexibility, right? It provides flexibility. Then we also see that this is responsible for the movement or allows entry and exit of substances, right? And in this case, we see that it provides rigidity. Yes, and it also provides mechanical support, right? This provides mechanical support. You write all these pointers, you will get 3 marks or 4 marks accordingly. Have you all do you want to take a screenshot, everyone? Quickly. You can take a screenshot. Very good. Cell wall provides rigidity. Exactly. Today's youth day also. Very good. It is, right? Yes, rigidity and protects from pathogens. Very good, Devanshu. Very good. Yes, all right. Okay. Ma'am, plasma membrane has proteins. It does have proteins. Plasma membrane has proteins. Vaibhav, happy birthday to you as well, bacha. Hope you had a nice day today. So this, of course, is the broad pointer, right? This is selectively permeable, completely permeable, found both in plants and animals, in plant and fungal. This is made up of phospholipid and protein, made up of cellulose. It has it encloses the cytoplasm. It encloses the protoplasm that is there. We see that it is flexible. This is rigid. So this is how you write the answer mainly, right? So are we clear with this? Are we clear? Vanshita, you probably missed the entire chapter explanation, but it's okay, we are doing it. Semi-permeable and selectively permeable, I have Anshika, yes, plasma membrane has proteins, right? Second thing, semi-permeable and selectively permeable. Selectively permeable, it allows only certain particles to enter or exit. Semi is with respect to structures, right? Yes, thank you Harshay, thank you so much. Now moving on to the next one. Another 5-marker question. Plant cell versus animal cell. You've been writing this from 8th standard, right? 8th standard onwards, you've been writing plant cell versus animal cell. Now you all tell me, ma'am, why is lysosome present if both remove toxic substances? Not exactly. Lysosome gets rid of unwanted substances. Toxic substances that are there is mainly removal of the peroxisome. So lysosomes not technically remove toxic substances, right? Yes, see, half yearly mein aaya tha. Like told you, all these questions are, our team has spent time to get amazing questions for all of you, right? Plant cell is dead. How is plant cell dead? Both cells are living only. Plant cells are not dead, bacha. No. See, very easy. Exactly. So plastids, they are present. Here plastids, they are absent. Cell wall that is there, in this case it is present. Here cell wall that is there is absent, right? Now size wise, if you see, we see that plant cells are slightly larger in size when compared to animal cells, which are slightly smaller in size. Now this of course is not part of your NCERT, but centrosomes is a type of cell organelle which is absent in plant cells, and centrosomes that are there are present in animal cells. What are centrosomes? Centrosomes are those cell organelles which are involved in cell division. And plant cells without centrosomes can regulate this one, right? Without centrosome only they are able to regulate their cell division. Locomotion is not absorbed. One more important thing. Here vacuoles are larger in size, while here in animal cells vacuoles are small or not present, right? Yes. Chloroplast point is plastid only in the beginning, right? That is already there. Anshika, you will not be able to talk about locomotion per se, right? Ma'am, plant can make their own food. Not all plant cells. No, plant cells which have chloroplast can only make their own food. So that will not be a proper point. Yes, dictyosomes are present in plants but not there in animals. If you given a point, you can write 14 differences. But for 4 marks to 5 marks, you have to write 5 points, right? So that is why you have to write these 5 points. One thing that is missing here is on vacuoles. So vacuoles present and is large in size. Here vacuoles are small or they might be absent, right? If you write this, you will get 5 marks each, okay? Now moving on to the next one. Question number 5, everyone. Diagram based question, okay? So you will get 2 marks for identifying this, 1 mark. So 3 mark question. Identify the given structure labeled X and Y. Yes? So see, I'm going to highlight it for you. X is inside part. Y are pointing to these foldings that are there. I mean, X is pointing to foldings. Right? See, who's telling? See, ma'am is not ignoring everyone. Exactly, ma'am is not ignoring anybody. Everyone has identified the organelle correctly. This is mitochondria. Yes. Cistern is not present. Cistern. Okay. What is present in mitochondria? What I've written as A or what I've written as B? What is present? Bhavya, vacuoles are storage sacs, right? Wherein we see that what proteins, amino acids, all of that is stored inside for usage. Yes? Very good. Now see, when you make spelling mistake, you see the difference. If you write spelling as this, gone, 0 you will get because this cistern is found in Golgi apparatus. But you all know that cristae is what is present. The inner foldings is what we call as cristae, right? Now if you write the spelling mistakes, gone, Damal, you have lost marks, okay? That's why spelling is very, very important, alright? Kriti, why are you saying I'm ignoring you, bacha? I would have missed your doubt. I'm not ignoring you. But let's focus on identifying X and Y. What is X? What is Y? X you have all identified, no? X is cristae. So these are the finger-like projections or the inner foldings. What is Y? Y is not specifying DNA. Y is not specifying ribosomes. But Y is specifying the matrix, right? So in the matrix that is there, we see that we will find the ribosomes and the DNA. Now what is its role? So 2 marks you've got here, okay? Those of you who told me cistern's gone, half mark, 1 mark gone there only. What is its role in the cell? What is its role? They're talking about the role of the cell organelle. Very good, Gia. Very good. That's how you have to write the answer. Mitochondria is also known as the powerhouse of the cell, which helps with cellular respiration or formation or synthesis of ATP. What is ATP? It is adenosine triphosphate, right? So that is what we mean by ATP. Structure of ribosome, you don't need to go into it, but it will look something like this, okay? It will have 2 units and it's a small structure. SK, you had a question on biosynthesis. I think what you are asking is, ma'am, where exactly does biochemical reaction take place? See, a lot of the chemical reactions that happen within the cell happen inside the cytoplasm, which is why that is the answer, right? So, of course, you need to write the answer in the given figure. X is cristae, Y is matrix, 2 marks here. What is the function? As we know, this is 1 mark. And you can talk about how exactly this helps, okay? Provides energy to the cell. We have 15 questions, 10 more questions and then Menti. Our aim is 8:45. 8:45, 8:50 is our aim to finish questions. Again, you have another diagram based question. Ma'am, plants have sedentary existence, so they should, no, because sedentary means they are not able to locomote or move externally, but a lot of internal movement takes place. And we know that characteristic of living organism is not just characterized by external movement, but internal movement. So you cannot say that plants, just because they're sedentary, are said to be non-living, right? Because characteristics range beyond just one thing. Okay. Now, moving on, which is the diagram that is given to us? What is the diagram? Quickly in the chat. What is the diagram that you see? Very good. This is a diagram of the nucleus. Now what do you need to identify? 3 mark question. So A is to figure out what is X and Y. What is X? X is, very good. X is X is nucleolus, right? What is Y? What do you see as Y? It's pointing to some thread-like structures. Exactly. Now, I want to ask you something. Inside, if you saw one rod-like structure like this, right? If you saw rod-like structure and this was marked as Y, what will you write? If in turn it was this, what will you write? What are you going to write? Very good. So now you know, right? If this rod-like structure comes, you should write chromosome. You write chromatin material, gone, damal, you will get 0 marks. Okay? But if it is thread-like, you need to write chromatin material. Okay? Very, very important. Yes? Very good, very good. Now they're asking what is the function of X. Okay? So now they're asking what is the function of X. That means what is the function of nucleolus. Very good. Ma'am, function is to control cell, DNA storage. Okay. I'm getting a lot of answers. But see, with nucleolus that is there, it is the site of, what do you say? It is site of ribosome synthesis, I would say. So the ribosomes that are there are produced by the nucleolus, and we see that, you know, I'll not call it as the brain of the nucleus, but their main function is production of ribosomes, right? Very good. rRNA synthesis or ribosome synthesis. That is how we will answer. So everyone quickly, function of X, that is nucleolus, is synthesis of ribosomes. 2 marks for this. I will not give 2 marks but 3 marks. 2 marks for identification, 1 mark for identifying this, right? Very simple and easy. Now moving on to question number 7. This is a 2-mark question on why are lysosomes known as scavengers of the cell. Ma'am, nucleoid and nucleolus difference. I have a question on that, Pranya. So when I do that question, I'll tell you. I'll do that. Because if I give you the answer now, then there also you know you'll have to tell me the same answer. And I don't want to give away the answer now. Okay, suicide bags also. Yes, exactly. Suicide bags. ये भी exam में आया था. See, I told you, no? Whatever questions we are picking. Simply, I can't see your comment, bacha. If you simply say you're ignoring me, then how will I know? Tell me what your doubt is. I'll definitely explain. Right? Because they destroy the cell. See, I want you to give me answers as how you will write in the exam. Take 1 or 2 minutes, it's okay, but try to type it. Because it removes, it releases chemicals. Yes. Good evening, Sani. Because whenever cell, yes, very good, Anshika, very good. Yes. So, first and foremost, right, we see that lysosomes are membrane bound sacs that contain digestive enzymes, right? So they contain digestive enzymes. Now the thing is, whenever, so 3 conditions are there. So whenever a cell, organelle or any other part is worn out, then we see that they get rid of it, right? By engulfing it by endocytosis. Now we also see that if any foreign particles enter, so if foreign particles enter, they will engulf and destroy it, right? And at times, right, if the cell is worn out or damaged, it will burst and destroy its own cell, right? So in all cases, why are we calling it as scavenger? We are calling it as scavenger simply because it is getting rid of all the unwanted substances, right? So that is something that you need to know. So when you're writing these pointers, please make sure that you mention all of these points as we have discussed. So if it is for 2 marks, you need to write only 2 points. But you need to make sure that you tell, right? You please make sure that you, when you are mentioning it, mention that they get rid of all the unnecessary particles, wherever it may be. Which is why in this case, we see that is there. Shan, I already explained, right? I have already explained Fred channel and all you don't need to know. You only need to know stroma and grana for in the case of stroma and grana in the case of NCRT. So don't worry. Ma'am, formation of lysosomes. Lysosomes are formed from the Golgi apparatus, right? So the enzymes which are produced or which are inside the lysosomes are produced by the RER, then they are sent to Golgi apparatus, and then we see that there is formation of lysosome. Yes, I hear Kriti also says hi, but Kriti, you had a doubt, so please, what is your doubt? And I think Gyana also had a doubt. So quickly everybody, whatever your doubts are, please ask me. Because if I miss your doubt, it doesn't mean that I'm ignoring you. Probably when I'm teaching you, because I'm looking at the camera, I would have not seen this, right? So I'm not intentionally ignoring anyone. My intention as a matter of fact is to make sure that after you watch this session, you're like, ma'am, fundamental unit of life is in my pocket, right? Okay, yes, Meenakshi, please ask me your doubt. Ma'am, middle lamella क्या होता है? See, middle lamella is a structure that is there, okay? So they are like these connecting structures which we find inside the plant, right? So that is what we call as middle lamella. Ma'am, sac means it's like a sac. Like how do you say? Like a imagine if you buy, how do I explain sac? Like potato sack, right? If you have like a pouch or a thing like that, which would look like this normally, right? So we see that this is a sac. That is what we call like that. Okay, ma'am, ye super ovulation not related to what we are learning about. What is endocytosis, right? What is endocytosis? Endocytosis is a process by which, because of the flexibility of the plasma membrane, we see that it is able to engulf, right? It is able to engulf or protect and take it in, right? So that is what we mean by engulfing. Krebs cycle, not part of the syllabus, so I'm not going to go into detail. Plasmolysis, I've already explained. Ma'am, what are centrioles? See, centrioles are these structures which we find inside the centrosome, right? And we see that they are responsible for producing the spindle fibers which are needed. Last of the session, can you make containing ribosome and DNA, and which not? Yes, done. Chromatid. See, chromatid is a structure which is formed during, like if you see, right, normally during cell division, we see that there is a duplicate which forms, which we call as sister chromatids. Okay. Krebs cycle, Shan, I will not be able to explain, bacha, because it is not part of it. Stages of cell division, I will do towards the end. Okay. Towards the end, all the extra out of NCRT syllabus I will teach. All right. Okay, let's come back to the next question. Question number 8. Easy peasy, 2-mark question. You will easily get in your pocket. Ma'am, please tell us questions on an NCRT point of view. These are some of the NCRT point questions that are there, Jhansi, but I will do it once again, okay? 2 marks. You have to, flagella is a whip-like structure that is there. Very easy this is. 2 marks. 1, you have to identify which is the powerhouse of the cell, and then why. Yes, very good everybody, very good. So the powerhouse of the cell is nothing but mitochondria. Why is it called as the powerhouse of the cell? Simply because it plays a key role in the production of ATP synthesis, or the production of ATP that is there. So this is how you write your answer. 2 marks for identifying the power, 1 mark for identifying power, 1 mark for writing the function. Okay, very good. Moving on to question number 9. Question number 9, sorry. Set up an experiment to demonstrate osmosis. This is a 3-marker question and very easy and important one, right? Very easy and important one. This is there in your textbook also. Yes? All right, very good, Anshu, very good. Yes. Raise in swelling. Now you can't just tell me, ma'am, raise in swelling. You have to tell me in the case of how you can set it up as an this one. Just give me one moment, everybody. Okay, very good. So when you're writing the answer, right? So in this case, you need to start, it says a lot of things. So in this case, so take what you can say is step number 1 is take a petri dish, right? And some dry raisins, right? Now first thing you need to do is to calculate the weight of, take 3 raisins and you can calculate the weight of 3 raisins, right? And then you soak it in water for 2 hours. Now what you observe is that, the observation is that the raisins appear swollen, right? And when we calculate the weight, we see that the weight has become more, which suggests that water that is there has entered from outside to inside. That suggests that respiration, it seems, osmosis has taken place. See, raisins are already no water. Don't then go and put it in salt water where further you will not be able to determine osmosis. So in this case, you will have to keep it in hypotonic solution. Here I think it is not OTP, it is OTP is your one-time password. I hope you are not asking me about that. ATP is what you should know in biology, adenosine triphosphate. Yes, exactly. But why is it called powerhouse? Simply because we have discussed it produces ATP. Now, same thing here. The weight that you take before putting in water is equal to W1. After putting in water is W2, right? It's just the weight, 2 kinds of weight. And ideally, for raisins, if W2 is greater than W1, then we see that in this case, osmosis has taken place. More specifically, water has entered into the cell. Alright? Ma'am, where is DNA in mitochondria matrix? Alright? Rhythm, I have answered. I have answered this. Okay? On what is W2. Moving on to question number 10, everybody. Nucleus and nucleoid. All of you can get this question for 3 marks again. And I'm sure all of you must be, very doubtful about this particular question. Ma'am, can we use cotton? No. Don't use cotton. It will not give you an accurate answer, right? Marshmallow, no, put raisin only. Why you want to use marshmallow and all? Use raisins, giving you decent answer, proper experiment. Ma'am, where does DNA? Dhana, I have answered, it is in the matrix. All of you need to pay attention after you ask your doubt, no? See if I am telling you, because most often than not, I actually answer most of your doubts. It's just that you have not paid attention that I have answered that doubt, right? Yes. Meenakshi, dictyosomes are structures which we find, right, in the plant cell. They are like these scattered structures around the Golgi that we find. Okay, for those of you, I know most of you have a doubt in this particular question, which is why I'll explain this part, okay? So now nucleus is a double membraned structure where the genetic material is found enclosed, right? That means if it's a double membranous structure, genetic material is found enclosed within. Now the nucleoid region is an area. Understand the difference. One is a structure, another one is an area, right? Exactly, Pranya. That's why I didn't answer your doubt then. So area where the genetic material is found concentrated. Okay, so if this is my cell, right, we see that this is my bacterial cell. In one part of the cell, the genetic material will be found concentrated, and we see that there is no membrane bound structure. Okay, so this circular thing I drew here, you will not find in this case, right? Now at the same time, right, the thing is, what we also observe is that nucleus is found in eukaryotic cells, while this we find in prokaryotic cells. Now everybody who had a doubt in this, are we clear? Are we all clear? What are genes? Genes are nothing but they are sequences, or I will sequences or they are fragments of DNA that code for a particular character. Are we clear with this? Are we clear? We don't need to talk about single life, double life. Let's all focus on academic life. Okay? Yes. RNA formula, we don't need to know Gyana, that we don't need to go in detail. Ma'am, enzymes present in nucleus. See, you have polymerase enzyme. There are so many enzymes. Helicase enzyme. Don't worry about all of that. You have 11th and 12th grade more to explore. But polymerase and helicase is one example. Okay? All right. Chat is disconnected. Ma'am, difference between organ and organelle, right? See, organelle is a tiny structure inside cell that works as an organ. Organ that is there is made of many cells that carries out a specific function. Are we clear? Are we all clear? Yes? Organ, organelle. Rajnandani is there here, but I need you all to listen to me. Okay? Nuclear envelope is nothing but the nuclear membrane, right? Difference between chromatic. What? I saw some plasma. Oh yes. Anshika, can I do that difference towards the end, bacha? I'll do that towards the end. Just remind me, okay, Anshika? Ask me towards the end, very important one. RER, SER, we have already discussed, right? RER, protein synthesis. SER, lipid synthesis. Simply, you are in class, no, bacha? You can go back and rewind, okay? So, this is about the main points of difference. This is how you will write. 2 marks means 2 points you write and you will get the answer, okay? Now, moving on to the next one. Distinguish between chromoplast and chloroplast, ribosome and centrosome. You have 2 marks for each. So, a total of 4 mark question. So, first, 4 more questions. See, I'll be fast, okay? I don't mind being fast, but I'll tell you, Niyati, everybody has to answer, right? Everybody has to understand. So everyone, I need your cooperation. Only then we'll be able to do. There is no point rushing me if your friends have doubts, okay? So be patient. Very, very important to be patient, okay? All right, okay. Now moving on to the next one. What is chromoplast? Quickly. What is chromoplast? Chromoplast is colorless or leucoplast is colorless? Okay, I am going to take you through the answer for this because I am not seeing any correct answers. You guys have not, you need to pay attention or spend some time in learning this, okay? So chromoplasts are basically, they have colored pigments in them, right? So we see that they have colored pigments in them. We'll not say they lack chlorophyll, but we see that they are normally found in petals, or we see that they are normally found in fruit peels, right? And we see that their main function is to, in the case of flowers, that is to help in attracting animals. Now chloroplast in the other hand is a type of plastid. So this is a type of plastid, this is a type of plastid, right? And we see that this is the one that gives chlorophyll. And we see that in this case, we see that they are mainly found in leaves, which play a key role in photosynthesis, right? Now when we talk about ribosome and centrosome, ribosome is mainly found both in animal cells, plant cells, and we also find in bacterial cell. But centrosome that is there is found only in animal cell. What is the function? Function that is here is simple. It is protein synthesis, right? And what is the function of centrosome? Function of centrosome is that they play a key role in cell division. So when you're writing the answer, these are the 2 things. I have already explained centrosome. Centrosome have centrioles inside them, and we know that during cell division they move apart, they produce spindle fibers, and they help with cell division, right? Now at the same time, yes, we also see that in the case of leucoplast, we see that it is a type of plastid which is colorless, and here they are mainly there for storage, right? That is the answer. Now moving on to the next question, okay? State two conditions which are required for osmosis. And to all my students who are asking me, ma'am, what is plasmolysis? This is also be there. Okay. Give me the answer and we shall move quickly. Yes? All right. So what are the two conditions needed? Prince Kumar, nucleus is found only in a eukaryotic cell, right? But in the case of prokaryote, we find, we mainly find this one. What is it? In prokaryote, we find nucleoid. Everybody, quickly. Very good, very good. Yes, all right. So as we know, right, for osmosis, what is it that we require? We need two solutions, right? So we require two solutions which have difference in concentration, okay, for osmosis to happen. Now at the same time, what we also require is a semi-permeable membrane, right? So for osmosis to happen, we require two conditions, or we require two solutions that are having difference in concentration, and we require a semi-permeable membrane. Anshu, I have already explained leucoplast. Please rewind and watch it, right? And simply regarding vesicles, they are small vehicles through which things are transported, right? Now in the case of plasmolysis, what is plasmolysis? When exosmosis takes place, right, or when a plant cell is kept in a hypertonic solution, right, it is kept in a hypertonic solution and it loses water through osmosis, we see that there is shrinkage in the cell, and it shrinks to such an extent that all the cellular components will go, and we observe that there is a gap. So we see that there is extreme shrinkage, and this is exactly what we call as plasmolysis, right? And such a cell is called as a plasmolysed cell. So I hope all of you are clear. What is exosmosis? Exosmosis is when water moves from inside to outside. That is what we call as exosmosis. All right? Very good, Archana. Very good. Okay. So this is how you will write the answer for this particular question. Right? Yes, bacha, we are studying bio. We are studying bio. 1 mark each you will get for this. Now moving on to the next one. Question number 13. Stay focused. All right. Give me the answers. We'll do the Menti. We eat food which are composed of all the nutrients such as carbohydrates, proteins, fats, vitamins, minerals and water. After digestion, they are absorbed in the form of glucose, amino acids, fatty acid. What mechanisms are involved in the absorption of digested food and water? Can you tell me? Ma'am, if we keep in hypertonic solution, the plant cell will get big. Yes, it will get big. It will swell. Okay? Simply, let's wait towards the end for membrane biogenesis. Protoplasm is the total amount of living substances that are there. Mahi, plant and animal tissue, we will be doing in tissues chapter. I am not dealing with it now. What is going to happen? Give me the answer to this. Ma'am, please explain this answer. I will explain. Yes. Guys, what are the mechanisms? You are all asking me doubts, but I am here waiting for all of you to give me the answer to this. What is the answer? I need you to stay attentive, right? I need you to give me the answer. You are all posting doubts, but none of you are paying attention to the question. If you don't know the answer, then you should tell me, ma'am, I don't know. You please tell me. Yes? They are asking, they have just twisted the question, but they are asking how does transport or how does movement of water take place? Are you all not aware of how movement takes place? How does movement take place? Exactly, it is through osmosis, right? Now when you talk about digested food, so now when you talk about glucose, amino acids, and if you talk about all of this, right, we see that it can happen either through some active transport. While in the case of water, we see that it happens through passive transport, because you know that glucose, amino acids, certain ions, all of them require energy, right? So we see that all of these require energy mainly. Now, of course, we see that active transport is how they are transported from outside to inside. Yes? Now, we see that for water, it is osmosis. So, digested food through osmosis, active transport, water through osmosis. They have just twisted the question and asked you. But simply, they have asked you how exactly is movement happening across the cell membrane. Are we all clear now? Are we clear? Are you understanding how to go about these questions? You all are excited to do Menti. I completely understand. But in your exam, nobody is going to come and tell you, Menti quiz is there, come, let's go. Nobody will ask you in your final exam. They'll ask you to write the answer for this only. At that time, you shouldn't be like, Arey, if I had paid attention to Aishwarya Ma'am that time, I would have done well in this. So you understand, right? This easily will come for 2 marks. 2 marks only this question will come, but aram se you will be able to get it. That is why when I tell you that, why is it that I'm doing subjective questions first? I could have done Menti first. But why am I doing subjective questions first? Simply because after I do Menti, you guys will be like, Arey, ma'am, why should I pay attention to subjective questions? I know, right? So that is why. The reason why I am doing this first is because you all pay attention till Menti comes and then we go on to doing Menti. But this is important at the end of the day. So don't dismiss this, all right? What is transport? Moving it from one place to another. Okay? Very good, Fatima. Very good. Alright, so this is the answer slide and 2 marks. Now, bachas, I will tell you one thing. If you spam the chat asking me for Menti, then unnecessarily delay will happen, right? So, we need you to stay attentive. Ma'am, can you explain this question? See, they have told you that we eat nutrients, we drink water, right? And we see that they are normally absorbed, or rather we know that proteins that are there eventually get broken down into amino acids. Yes, but they're saying how does amino acid enter into the cell? How does it enter? We know that all these ions, digested substances, they all enter in the form of or enter through active transport. What does active transport need? It requires ATP, right? So that is what we mean by it. Simply, I have already explained centriole. Rewind and watch the part, okay? Now moving on to question number 14. Second last question, okay? Second last question. Now, how do substances like carbon dioxide and water move out of the cell? 2 marks. Very simple and easy, right? Very simple and easy. Now, how do substances? See, through plasma membrane, I agree. But how exactly? Carbon dioxide and water. You cannot tell me passive transport will get half mark for this answer. Very good, very good. So, this is of course through diffusion. This is through osmosis. You have to specify that, okay? So, diffusion is the free movement of molecules from a region of higher concentration to a region of lower concentration, right? And gaseous molecules move in that manner. While in the case of osmosis, we know that it is movement of water molecules from a region of higher concentration to a region of lower concentration through a semi-permeable membrane. So, you need to write the definition, 1 mark each for writing this and identifying it, right? So, that's how you will get your 2 marks. Now, last question, okay? Last question and then we'll do Menti, okay? Last question. In brief, what happens when the plasma membrane of the cell breaks down, okay? This is a 2 marker. I will give you 4 marks for this particular question. What is going to happen when the plasma membrane of a cell breaks down? Cell will die, okay. Why will cell die? Why will cell die? Why will cell get damaged? Yes? Yes, yes. Okay, I'll be doing improvement in food resources. Don't worry. That will be in the 3rd week or 4th week, I think. Tissues, mostly Ankita ma'am will be taking the live class. Very good. Ma'am, they'll digest its own cell. Guys, don't overthink the question. Don't overthink it. Bring it down. Bring it down. is the role of the cell membrane? It is to keep all the cytoplasm, all the cellular content and everything inside. If this damages, what will happen? All that is inside will go out, right? All that is inside is going to come out and eventually the cell will die. Because we know that all these things come together and they make it up, right? So they will not digest anything. If it breaks up, what will happen is all the cellular contents will go and then the cell will die, right? So that is one thing. Now moving on to the next one. When a red blood cell is kept in a concentrated saline solution or concentrated salt solution, what is going to happen? You have an RBC and you are keeping it in a hypertonic solution. What is going to happen? Cell will swell here. Will cell swell or cell will shrink? Cell will swell for hypotonic. Guys, cell will not swell. Cell, water moves outside, so the cell will shrink. See, concentrated. Pay attention to the question. Concentrated salt solution. What do we mean by concentrated salt? That means more amount of salt, very little water. That means that when I keep an RBC, water that is there will move from inside the cell to outside. So that means that it is going to shrink. Okay? When will cell swell? When outside is more. If it was a dilute solution, it would have swollen. But if they say concentrated, know that it is going to shrink. Are we all clear with this? Are we all clear? Normally we use it for water prana, but in some cases also we may use it. So that's why we say for solvent molecules, but mainly water, right? Yes, all right. Yes, perplex gamer, that is the answer. Now everybody very quickly. This is the answer to both the questions. You will get 1 mark for justifying this, or since in my case I told you 2 marks per question, 2 marks for justifying what's going to happen, right? So with this we have finished chapter summary. We have finished 15 objective questions, right? So everybody, before we get into Menti, I hope all of you have hit that like button and all of you have subscribed to our channel. Now, if a lot of you are very new to the class and you have been sitting through my class for 3 hours, you will understand the quality of education that we give here at BYJU'S. We don't compromise with the quality at any point, and we make sure that a lot of you that are there, we make it a point that we help and we try to clarify as many doubts as possible, right? So we take your classes, we take your exams, we take your understanding very, very seriously, okay? Which is why I hope all of you have subscribed to the channel. Now, I can see I have close to 130 plus live watching in my class, and I hope that we hit a quick 250 likes in our video. Yes? How many likes do we have? 240. We need 250 likes, everybody, very quickly. And I know you can break this record of 250 very fast, and then I'm going to get on to Menti, which we will finish quickly in half an hour. Yes? Exactly. Now somebody was telling, ma'am, are these questions enough? No. You need to do everything. You need to do NCRT exercise, exemplar, and then that will help you. Ma'am, where is sir? Shiva sir is sitting here, right here. Okay. Let's break the record, everybody. Let's break the record. Okay, ma'am, it crossed 250, 262, 265 we shall touch and then go. Shall we take? Is it 265? I need 265 and then I'll go. Rupesh, if you tell me, ma'am, 265, then see, I have sir who's going to tell me otherwise, okay? 267. Here's the Menti code, everybody. Go to www.menti.com and type in the code 36085781. Everybody quickly go to 3menti.com. Yes? Yes, all my efforts and all your efforts have given you quickly. Go ahead and you can do Menti, right? Now Menti may I need to see all of you getting answers. So much enthusiasm for Menti. No, now I will see in Menti. Yes, all right. So can we freeze the screen, please? Okay. Let's have the screen frozen so that they can see the code. And once I see that all of them have joined, we can get started. Ma'am, what are the properties of amoeba as eukaryote? They have a well-defined nucleus. That is why we call it as, well-defined one, right? Yes, my ultimate target is to have 500 likes on this video. So we can do that. Now everyone, quickly. Jaldi, 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 we shall go. I have kept the code on screen so that all of you can join and then we'll get started. Yes, Simpy, done. Done. Promise. Very proud of you, Prabhu. Very proud that you've done well in your bio exam. 270 likes. That's amazing. We can hit 300, that means. Thank you, Monish. Very sweet of you to say that. Very sweet of you to say that. So guys, I can see that around 95 of you have already joined. Shall I get started? Simpy, if you see in the channel, there's a community post which will tell you when exactly we are going to air which, when exactly the class is going to be there. The dates are there, bacha. You can go ahead and look at it, okay? All the dates are there. Yes, goat, teddy bear, alien, everything is there. Let's have a look at the first question. Question number 1 on your screen. The idea of omnis cellula e cellulae, which means that all living cells arise from pre-existing cells, was given by who? Robert Brown, Purkinje, Rudolf Virchow, or Schleiden? This is part of cell theory that we discussed, right? Very easy question, everybody. Very easy question. We have already discussed. Halwa question, no? I did wrong in hurry. Saurabh, it's okay, but next time pay attention. So Rudolf Virchow was the one who contributed to cell theory, where he added this point that all living cells arise from pre-existing cells. Purkinje coined the term protoplasm. Robert Brown discovered nucleus, right? And Schleiden contributed in the case, or Schleiden contributed to the cell theory. So the correct answer here is option C. Ma'am, where is sir? Bacha, which sir? If you are asking Shiva sir, then he is sitting here. Saurabh sir has gone home, or I think he's outside. I don't know. Let's see. Are we clear? Shall we move on to the next question? Question number 2, everybody, on your screen. Leaderboard shall come to the shall come after every 5 questions. My BFF, my best friend, is taking class for class 10. Okay? Yes. All right. Moving on to question number 2, everybody. Question number 2 on your screens. Cytoplasm is what? There are 4 statements. Pick the correct answer. It is a unit mass of protoplasm. Protoplasm excluding plasma membrane, protoplasm excluding plasma membrane and nucleus, or protoplasm excluding plasma membrane and cell organelles. Very easy. Very, very easy. Pay attention. Menti code 36085781. Very good. Now, how many of you liked this question? How many of you like, ma'am, even if I got it wrong, this was a very nice question. How many of you liked it? You enjoyed this, no? It's a good question. See, you learn everything in one way, okay? One way only, you will keep learning it. But this way around, what helps you is to look at it at a different perspective, right? So in this case, when we talk about cytoplasm, we know that it is the jelly-like fluid which is between the plasma membrane and the nucleus. And when we talk about protoplasm, it includes your cytoplasm, nucleus, cell organelles and plasma membrane, right? So the correct answer here is option C. Ma'am, cytoplasm may cell organelles. In the cytoplasm, the cell organelles are found suspended, right? But cytoplasm is not a part of it, okay? All right. Now moving on to question number 3, everybody. Question number 3 on your screens. Be ready. Don't tell me, ma'am, I did not get to write answer. No, I did not get time to write my name. So quickly, everybody, be ready. Okay? All right. Now moving on to question number 3. Mitochondria. See, so many questions on mitochondria has come. Everybody should get the answer for this. Okay? Mitochondria is the site of aerobic respiration, Krebs cycle of aerobic respiration, glycolysis of aerobic respiration, or anaerobic respiration. This is a tricky one actually. It's not direct. This is a little tricky. See, aerobic and anaerobic is a concept that you learn mainly in your 7th standard, 8th standard, right? Very good, very good. Anshu already gave me the answer. Some of you have already got this part correctly. So well done. Now, I'll tell you one thing. Ma'am, I did not understand glycolysis. See, what is respiration, right? So, respiration is nothing but the breakdown. It is breakdown of glucose in order to release energy, right? So this is what we understand by respiration. Now respiration can happen in an aerobic manner or it can happen anaerobically. So if aerobic is there, and this you learn in 10th standard also, okay, so don't worry. Aerobic that is there is in the presence of oxygen. Now this particular thing takes place in, this particular thing takes place in mitochondria. Now Krebs cycle that is there is a part of aerobic respiration, but you see Krebs cycle is only one part. So we cannot really say that this is the answer, okay? And for Krebs cycle, half of it, like there is some steps which are happening outside mitochondria, some which are happening inside mitochondria also, okay? That's why. And Krebs cycle you don't need to worry. Now what is glycolysis? Glycolysis is nothing but breakdown of glucose into an intermediate product which is called as pyruvate, alright, and this takes place in the cytoplasm of the cell, alright. And anaerobic respiration, we know it is not even related to mitochondria, right? So correct answer here is option A. Now moving on to question number 4, everyone. See, in Menti also very good questions are there for proper conceptual understanding, right? So take this as a challenge and let's have a look at question number 4. Yes, question number 4, everyone, on your screen, right? Genetic material of a eukaryotic cell is present where? Nucleolus, nucleus, nucleoplasm, or nucleoid? Very quickly. That is amazing, Simpy, that you found interest and you subscribed. You tell all your friends also. BYJU'S 9 and 10 is amazing. You should subscribe here only. Yes. Ma'am, what is diploid and haploid in cell division? Diploid is when 2 pairs of chromosomes are there, like in 46. When 46 chromosomes are there, you have 23 pairs. But if you have only 23, half or one set of chromosomes or half pair, the correct, we call that as haploid. Guys, everybody who got confused here, what happened? Genetic material or DNA. Where do we find this? Where do we find DNA? Right? We find it inside the nucleus. Nucleolus we will not find. Nucleolus is responsible for ribosomes, right? Production of ribosomes. What is nucleoplasm? Jelly-like substance. What is nucleoid? Nucleoid is when the genetic material is found concentrated in prokaryotic cells. Everybody revise this part because over and over today all of you have been making mistakes in this part. Make a star mark and tell yourselves you have to revise this. Apican blossom and honey, Kamu, all of you, no discussions, stay on Menti, okay? All right. Moving on to question number 5, after which we will have our first leaderboard. Code is 36085781. Question number 5, everyone, on your screens. A cell, when it's placed in a solution, the solution will swell up. So this is cell will swell. When is cell swelling? I have been singing this all day today, okay? When is cell swelling? Cell will swell when? Hypo, hyper, iso, both A and B. I have my fingers crossed that you get this answer correctly. You have to make me proud, okay, by getting this answer correct. I need to see majority here. Majority is what I want to see. Very good. Very good. I am so proud. I am so happy, right? Now, most of you have got the answer correctly, right? Yes. Ma'am, I learned cell. Hypo means hippo. That's also a good way of remembering, but no, that's also a little bit on shaming, body shaming. So let's not go there as well. Cell will swell when kept in hypotonic solution, right? So when in a hypotonic solution, it will swell. In hyper, it will shrink, okay? Hyperness will make you shrink. Hypo means swell, right? And yes, if you want to use what Epic has said, you can use that as well, okay? Ma'am, Menti आज नहीं होगा, यही Menti है. Code is 36085781. Join quickly. Now, before we go to the next question, let's have a look at the leaderboard after the first 5 questions, right? Yes, let's have a look. So somebody who forgot to tell their name or has written their name as name is the fastest, followed by Krish, Navansh, Himanshi, Garvit, Apshita, Jia, Casper, Roshandeep and Jayashri. Well done everybody, well done. I will give you 5 stars for your amazing performance and 5 stars to all of you. Another 15 minutes may we'll wind up. 15 minutes Menti and we're going to wind up the class. Okay, and end may I'll give you some tips and tricks. Benam, yes, exactly. Who is name? Name apna yeah, name apna name bata dena please. Very good. 16th, 83, 28. Good positions. Good. Well done. Yes. Well done everybody. Well done. Very proud of you. So now let's quickly move on to the next set of questions. Right? Quickly everybody. Next set of questions on your screens. Be ready on your Menti screen. Yes? All right. Okay. Question number 6 everybody on your screen. For those of you who are very new to my class, welcome everybody. Okay, this organelle which is not present in a human cheek cell is which one? Nucleus, plasma membrane, mitochondria, chloroplast. Pankaj, I told you another 20 to 20 minutes I will finish. 20 more minutes. This was supposed to be a 4-hour marathon. No, that's why. Easy peasy, halwa question this is. Halwa, double halwa question. Very good everybody, very good. Most of you have got the answer. I don't know why all of you got confused here with nucleus, plasma membrane, mitochondria. I think some of you had misclicks as well. When we talk about human cheek cell, we are talking about an animal cell, right? So when we talk about an animal cell, we know that it is mainly chloroplast, right? So answer here is chloroplast because chloroplast is something that is not present in animal cells but only in plant cells. Now moving on to the next one, right? Question number 7 everybody on your screens now. Find out the incorrect statement. I am highlighting this. What is incorrect? You have 4 questions which are 4 statements out of which you need to identify the incorrect one. Take your time and answer this. Ma'am, I have doubt. Yes, please ask me your doubt. This is super easy, guys. Very good, everybody. Very good. See, Golgi apparatus is involved in the formation of lysosome. This is true. Now, before I go ahead, how many of you read this as identify the correct statement? How many of you read the question as identify the correct statement? Ma'am, what are all the organelles in a cheek cell? All the cells that you will find in a normal cell organelle, you will find in the cheek cell, Rupesh. How many of you read it as correct? I know there will always be some few people who read it that way. See? That's why mistake happened. Yes, exactly. If you've read it, then whoever has marked this, you have marked it because you thought it was correct. Incorrect is when you say cytoplasm is called protoplasm. We know that cytoplasm and protoplasm are different, right? Protoplasm that is there is the total living substance. Cytoplasm that is there is the jelly-like fluid. Very simple and easy. Read the question first. Don't run to answer. Okay? Ma'am, explain question point number 3. Mitochondria is said to be the powerhouse because ATP is generated. I think Menti did not continue generated as the word, right? But that is the answer. Question number 8, everybody. I'm going to pick up the pace now. Question number 8 on your screens now. Quickly. Which of the following is an incorrect pair? Look at the word. It says incorrect. I am circling on this. Chloroplast, kitchen of the cell. Mitochondria, powerhouse. Lysosome, secretory. Nucleus, brain of the cell. What is the correct answer? This is so easy. Even if you don't know, you will be able to find the answer to this. Very good, everybody. Very, very good. Now here again, I can see that some of you here have marked chloroplast as kitchen. This is not incorrect. This is correct only. Lysosome is not secretory granule. Lysosome is what? It is a suicide bag, right? Or else lysosome is said to be scavenger. But secretory granule is something which you cannot really talk about lysosome, right? So the correct answer is option C. Now moving on to question number 9, everyone. Question number 9 on your screens now. Misclick ho gaya. Koi nahi. Everybody, let's have a look. Okay, Vanya, sometimes I think it's difficult, right? Sometimes it happens. Menti is a third-party platform. Amoeba acquires its food through a process termed as what? Exocytosis, endocytosis, plasmolysis, or both. Ma'am, they have secretory enzymes. They never secrete enzyme, Himanshi. What do? They will engulf it, right? They will engulf it and they will break it down within themselves, or in extreme cases, they will burst open. They are not secreting any enzyme, okay? So that's why it's not the incorrect pair. Very good. Amoeba acquires its food by engulfing, which I also gave in my explanation, by the process of endocytosis. Now moving on to question number 10. See, we are saying how it obtains its food. Obtaining food or getting the food is by endocytosis, right? But if you talk about the whole process that is there, we see that, what do you say? If you talk about the whole process, yes, exocytosis is a part. But when you just talk about obtaining and getting the food, that is endocytosis. Question number 10, everybody, on your screens now. Select the odd one out. Again, take your time with this, okay? The movement, there are 4 options. You guys read and get the answer correctly. I am not reading the options. Yes. Oh, that's how sweet. Can we refresh the chat? Okay. Odd meaning which is standing out, right? Which is different from the other. See, what is the first one? Movement of water across the semi-permeable membrane is affected by how much substance? That means movement of water depends upon concentration. Yes. Membranes are made up of organic molecules. That means your cell membrane is made up of proteins and lipid. Yes. Molecules are soluble, right? Molecules are soluble in organic solvents. So mainly if you talk about solvents, molecules are soluble and they are able to move like your water. Plasma membrane consists of chitin. No. Even if you don't know the rest of it, plasma membrane is made up of what? They are just telling you here only protein and lipid. Then why will we say that plasma membrane is any different in plants? Which is why the correct answer is option D. It's okay, it's mainly about how you learn it, right? Yes, after doing Moral of the story is what? I got a very interesting thing there. Never trust the chat. Oh, okay, fine. Yes, that is the moral of the story. Now let's have a look at the leaderboard, everybody. Second leaderboard for the day. Let's see what the leaderboard has to say. Okay, I can see Krish is on top now. Himanshi, then Jia, Apsita, Name. Then I can see Tal, who's the fastest. Casper, Garvit, and Vaishu. Well done, everybody. Well done. Very proud of all of you. Arpit is like, ma'am, I went from 47 to 77. It's okay. It's how you learn. See, at all times we tell you that if you make any mistakes today, even if you make the same mistake 10,000 times, it shouldn't be that you shouldn't be making the same mistakes because you're not learning from it. If you make a new mistake, it's fine. But every time you make a mistake, try to figure out what you did. Because here is the place we want you all to make mistakes so that when you go to your exams, you will never make those mistakes again. Right? So that is the reason behind it. Now the last set of 5 questions, everybody. Last set of 5 questions. Yes, and we will wind up our class with some tips and tricks. Okay? So question number 11, everybody, on your screens. Match the following items from column 1 to the ones in column 2. See, whenever match the following is there, I always tell you, right? I always tell you how to go about matching it. And I'm going to keep it in my phone as well so that we'll be able to match it later. Otherwise, I'll forget what is what. Take your time and match. Don't rush with it. Very good, everybody. Very good. But I can see some of you have made silly mistakes. Now, the first one that we had was SCR, right? Now, we know that SCR is mainly responsible for lipid synthesis, and we also know that it plays a key role in detoxification, right? So, we see that the first one here is detoxification. Now, the next one that we have is lysosome, okay? So, we have lysosome. What is lysosome? Lysosome is also known as the suicide bag of the cell, right? So, that is what the match is. Now, next we have C, which is the nucleoid region. Now, I feel like this is where you guys would have made the probable mistake, right? Now, we know that the nucleoid region is something that we find in bacteria, okay? Now, what are we left with? We are left with food vacuole. Now, food vacuole formation takes place in an organism like amoeba, right? So, we observe this in amoeba due to endocytosis. And of course, last what we have is chromatin, material and nucleus. Nucleolus. And we know that this is something that we find in the nucleus of the cell, which is why this right here is the correct answer. So, SCR is smooth endoplasmic reticulum. So, are we now all clear with this? Are we clear? Ma'am, iske baad mein ek error. Oh, okay. No problem. It happens. All right. Question number 12, everybody. Question number 12 on your screens. Very quickly, be ready to give the answer to me. Yes, I know for this, actually, they should have made it 40, it should have been 40 seconds, but it's okay. It's fine now. Now, moving on to question number 12, everyone. Which of these is not related to endoplasmic reticulum? Okay, you will have 4 statements. Which of the following? Use. If you don't remember all the functions of ER, the odd one out is very easy. It is so easy, so direct, you will be able to get it like this. Very good, everyone. Very good. Which of it is not related? Guys, I know that this bunch of people have read as which of it is related to endoplasmic reticulum. How many of you read it this way or how many of you had misclicks, right? Because Energy generation is mitochondria, right? Energy generation is mitochondria, which is why, ma'am, wrong answer. Exactly, which of these is, if it is not related? No, exactly. So in this case, it's very simple. Which is not related? We know that it acts as a transport channel or transport channel in various ways, right? And it can be at times site of biochemical activity. Synthesis of protein is a biochemical activity. Energy regeneration is far off from it, right? Which is why this is the correct answer. Now moving on to question number 13. Yes, very good. Question number 13, everybody, on your screens now. Which of the following statement is incorrect with respect to cell division? Read it carefully. Incorrect. Two types of cell division is there. In mitosis, daughter cells have half the number of chromosomes. Meiosis is a type where it involves two divisions. New cells are formed. Easy peasy. Easy peasy. But if you don't read this correctly, or if you have not gone, because see, today was a summary, which is why I did not teach a little in detail. But if you have watched Ankita ma'am's classes on cell division, you will be able to answer this. Or if you have not watched it yet, you go and you check it out. Alright? Yes, I knew some of you would have either read it as what is correct about cell division. So we know that cell division is of two types, mitosis and meiosis, right? Now mitosis is often also called as equational division, right? Why are we calling it as equational division? Because if the mother cell has 46 chromosomes, like in the case of human, right? We see that the offspring will also have 46. It will not have half the number. Half the number is for meiosis, okay? And meiosis has two stages. It has meiosis 1 and this has meiosis 2. Okay, so there are two stages. And why do we need cell division? To get rid of old cells, to make sure that new cells are there and injured cells are replaced, right? So that is the reason behind it and why the answer is option B. Now moving on to question number 14, second last question, right? Second last question that is there. In your question, it's going to be there on your screens. Tomorrow actually for grade 9 there is no class, right? The SST class which was ideally scheduled is moved to the next week. So tomorrow for grade 9 class is not there, but you will have it on Saturday with Khusbhu ma'am, right? So Saturday you have class with Khusbhu ma'am. So tomorrow you can use your time wisely and prepare well. So which of the following movements across the cell membrane requires energy? Very easy. Which requires energy? That means 3 out of these 4 options, no energy is needed. Very good, everybody. Very good. Passive transport, no energy. Osmosis and diffusion are passive transport, right? So correct answer is option D. Now moving on to last question. Last question of Menti. Yes, last question of Menti. Let's have a look, everyone, on your screens now. Which of the following is not a function of the vacuole? Yes, which of the following is not a function? Storage, providing turgidity and rigidity to cell, waste excretion, or locomotion. What is the correct answer to this? And those of you who have just joined the class, right? Please make sure that all of you don't forget to hit that like button. Very, very important to like and subscribe to the channel. Very good. Now I know some of you, how many of you read it as, ma'am, what is a function of vacuole? How many of you read it this way? Ma'am, can you explain once again? Karan, which one? Which one do you need? Locomotion kya hai? When we go from point A to point B, it is called as locomotion, right? So in this case, storage is a function of vacuole. Providing turgidity and rigidity. Turgidity especially because it stores water also. That is also something which we find. And in the case of amoeba, it is also necessary for waste excretion, right? Which is why the correct answer is locomotion. So let's see what the final leaderboard says. Yes, final leaderboard. Ma'am, storage one is me, misclick. It's okay, Epic, no problem at all. Well done, Krish. Well done. Krish has 13,492 points, followed by Apsita, Himanshi, Boss, who is the fastest, Name, Shrishti, Nakshatra, Sachin, and Garvit. Very good, everybody. Very good. And very proud of how hard you have all worked, right? See, today was not an easy class. We had almost, I would say now 3 hours and 40 minutes of class, 3 hours, 30 minutes because I started a little late. But I'm very proud of all of you for how much you were so attentive today, right? You were very attentive, you were very interactive, and you went ahead and you were asking me all of your doubts, right? Now, for those of you who want certain tips and tricks, so if, Shiva, if you could just freeze my screen for a minute, right? Yes. So for those of you, right, if you feel like, ma'am, we did this, but if I need to prepare for maybe, fundamental unit of life, right? If I wanted to go ahead and if I have to prepare, how do I go about this? Yes. So I'm going to give you some quick tips and tricks on how to revise. Also, I will not spend too much time, 2 minutes of my time in telling you 5 easy tips and tricks in helping you study, right? Yes. Those of you who are hungry, you can eat in my class also, like you can eat and you can ask your doubts as well, no problem at all. Yes, and Priya, this is your first class on YouTube, and of course, right, if this is the first time that you are here with us, I'm very happy. I hope all of you have liked this particular class. You enjoyed today's class. For all of you who are very new to your first class on YouTube, let me know if you enjoyed today's class. Did you enjoy? Did you find this totally helpful? Let me know very quickly in the chat. Yes? Okay, can I switch? Okay. All right. So as they set it up, right? Is it set? No problem. It's okay. So as we set it up, yes? So everybody, as we set it up, because Shiva sir is working very hard for all of us, right? So as we all know, I hope all of you have also registered by giving this quiz, right? I hope all of you here have registered for BYJU'S Quiz Masters. You know that BYJU'S Quiz Masters is happening on 28th of January 2023 and registrations are open for all of you. So everybody please go ahead and register. Yes. So the link is there in the description below and Tarana ma'am will be taking it. So do check it out. What is Menti? Menti is nothing but a live platform where we play. So what did we do today? We have covered all these 3 things, and last we are going to look at 5 hacks that will help you remember this chapter best, right? So for structure function of cell organelles, what I would recommend is that you make a tabular column and you study this way. So when you're talking about chloroplast, go ahead and check this way out. So I have one table for structure, one for function. And in many places, there are many commonly asked difference questions, right? So if you think about it, difference between nucleus and nucleoid, prokaryotic, eukaryotic, plant cell, animal cell. So you can make a table in this way and you can check it out. And it's always important that you have this compare contrast, right? Compare and contrast and study. This makes revision very, very easy. Now the third one, of course, is to make flow charts that will help you, right? So you have prokaryotic cell, eukaryotic cell, under which you have plant and animal. And of course, practice diagrams because they will be very important. So plant cell diagram, animal cell diagram, structure of maybe, chloroplast, mitochondria, all of these things will come. Yes? And last but not the least, try to use the prefix and suffix. So wherever endo is there, that means inside, right? Endosmosis, endocytosis. Exo means outside, exosmosis, exocytosis. All of these things will help you out, right? And of course, with this, we have looked at the 5 hacks. Now, where will you find these notes? You will be finding it on our Telegram channel. So the link is there again in the description box and you will be able to know. So, ma'am, table for protein. Okay, yes. Now, there are 2 things that I will take. Any more doubts? For those of you who are done with this, it's okay. For ribosomes containing, for organelles containing ribosomes and proteins. See, ribosome plus proteins, or which can synthesize their own proteins, include chloroplast and mitochondria, right? But those which do not have this include your endoplasmic reticulum, right? Or I would say this includes your Golgi, this includes your this includes your lysosome, right? All of these are examples of the same, right? Now at the same time, another one is osmosis and diffusion. Yes, so we have osmosis, diffusion, and we have active transport. I'm writing it as AT. So first one, osmosis, is mainly for water molecules, this is mainly for gases, and this is mainly for nutrients and ions, right? Now how does this happen? Semi-permeable membrane is needed, semi-permeable membrane is not needed, again semi-permeable membrane is not needed. Now in this case, energy, right? So energy is not needed. Here also we see that energy is not needed. Here energy is needed. So everybody, you can take a quick screenshot of this. This will definitely help you out, right? So everybody, I hope all of you enjoyed today's class. If you enjoyed today's class and if you have any more doubts that I have not answered, let me know in the comment section below. I will definitely answer. And of course, if you enjoyed today's class, you have to let me know that, ma'am, I found this class amazingly helpful, and you should tell me that as well. And everybody, don't forget to like, share and subscribe to the channel. And I will be signing off. Any more doubts that are there, let me know in the comments of this video and I will help you out. Up until then, everybody, please take care. Lots of love to all of you. Hoping to see you soon again. Bye bye and have a nice day.